I hope I am audible. Let me just check it, then I'll start off the class. Yes. So I am audible, and Anish, hello. Rida, hello. Guys, I hope if at all you had your math examination, it was fine. Okay, it was good. Okay, so listen to me very carefully before I start with the final revision which is there. It is going to be fast, but before I talk about that, you are going to write biology KCET examination. Suppose you have the paper there with you, and you find that first set of questions are difficult. Do not get demotivated. What you have to do is skip those questions, as in, for example, first question is difficult, second question is difficult. What you can do is skip the first 10 questions and move on with the 11th one. First 10 questions you move on with the 11th one. Probably you might be able to find an easy patch out there. So there might be easy questions which are clumped up together. Make sure you target that. Once you do that, basically you will get the confidence. If not, still just make sure that this is just an examination and I'm just trying for it. Do not be so sad. Do not get demotivated there. You will not be able to perform well. You might lose the chance of doing better. Okay, you have the potential. Believe in yourself and just go ahead. That is the first thing that I want to tell you. And please remember that in biology you make silly mistakes by not reading the question well. So read the question well and then only read the answers well. That is the options well. Okay, so question first and then the options. Properly take your time, you will do good. Okay, hello everyone. Good afternoon. So this is just a final revision for everyone out there, and I hope this is definitely going to help you in some ways. Definitely questions are not going to be repeated as such, or it's just like a revision of the concepts which are there. It is going to help you. Yeah, starting with the PU1 concepts which are there, because PU1 is something that is left untouched. Let's go ahead to the first question. In which type of vascular bundles are xylem and phloem present on the same radius? That means the xylem and the phloem which are there, they are present together like this. In which type of arrangement do you see that? Radial, closed, conjoint, exact. Always remember, if at all you have an arrangement like this, you will call this as conjoint. You might have heard about conjoint twins, right? So similarly over here, this is going to be conjoint. And this is also what? Conjoint itself. Here also phloem and xylem are together only, but there's a slight difference. Please understand. Here between the xylem and the phloem, there is nothing. But here you see something that is known as cambium. Okay, there is something that is known as cambium. If at all there is conjoint arrangement, but you have cambium in between, you call this as open setup, and this is found in the case of dicots. So conjoint as such, something like this, is found in the case of stem only. It is not found in root, only stem. Okay, conjoint only in stem. Now how do you understand whether it's dicot stem or monocot stem? A dicot one will be having a cambium. You call this as open arrangement, conjoint open arrangement. A monocot one will not be having cambium, which is why you will call it conjoint closed arrangement. So stem, okay, this is all stem, this is dicot stem, and this is going to be monocot stem. What about the root, ma'am? Root, R for root and R for radial arrangement. Here the xylem and the phloem are going to be in alternate pattern. Okay, they are going to be in alternate pattern, xylem, phloem alternatively. They are not going to be conjoined. Radial root, always remember. Now you have dicots and you have monocots. How do you understand that? Dicot one which is there, okay, dicot root will be having cambium just like this, right? And monocot root will not be having cambium. Have this understanding and you might be able to solve at least a few questions if at all that comes. Okay, if that comes. So I'm just repeating, radial for roots. If cambium, if it is mentioned that radial open, just understand open means dicot. Radial closed, that means it is going to be monocot root. If at all conjoined, it has to be a stem. How do you understand dicot stem or monocot stem? If cambium, dicot stem. If no cambium, monocot cambium. Monocot stem. Cool. So that's all. Next one. Functional unit of contraction is known as sarcomere. What do you call it? Sarcomere. And sarcomere looks like this, Baba. Look at it. Over here you have A band and you have I band. A band consists of the thick filaments. What do you call them? Myosin. What do you call them? Myosin. They are thick filaments. Okay. On the other sides, what do you have? You have actin. Actin are what? Actin are the light filaments out there. So actin and myosin together forms all the things out there, and this is is known as sarcomere. Sarcomere unit is a unit which is there between two Z lines. Two Z lines are there, and in between that all of this is happening. Here you have A band where just myosin is there. You have I band. What do you have? You have actin. When the muscles contract, what is going to happen? The Z lines which is there, they are going to come close like this. This remains intact. This comes closer. When this comes closer, what is going to happen? The I band which was utilizing this much amount of space will now utilize less space. So we say that during contraction, I band get reduced. What about the A band, ma'am? It stays as such. It is going to stay what? As such. Always remember. I'll repeat it once more. Listen to me. A muscle's contraction unit is known as what? Sarcomere. It is the unit between two Z lines out there. In between myosin, towards the side you have actin filaments. This band is known as I band. The middle band is known as A band. A band remains as such. No change is going to happen. I band is going to get reduced only when contraction is going to happen. This is one very important thing. So the functional unit of contraction is what? The portion of myofibril between two successive Z lines. That's all. Guys, I hope you're understanding. I am going fast just for you to have a quick revision. That's all. Now, identify the incorrect statement with respect to the rules of binomial nomenclature. What is binomial nomenclature? Binomial nomenclature means two names will be there for an organism. For example, Homo sapiens. Homo is what? Our genus name. Sapiens is what? The species name. So always understand that over here, the first word in a biological name is going to be genus, but the second word is going to be what? Species out there. And it is always going to be in Latin. If not in Latin, they will convert in Latin, but definitely it is going to be there in Latin only. So Latinized or derived from Latin irrespective of their origin. And when you are writing it, you underline it. So whenever it is handwritten, you keep the name separately, the genus name and the specific name, and basically you underline it. If at all you're printing it, you're going to print it in italics, and that is to indicate what? That they have a Latin origin, not a non-Latin origin. So please understand, this is very, very important question. A question came in 2025 itself. If at all you're lucky, you can get one more question on binomial nomenclature, but just understand. So biological names are printed in italics to indicate their non-Latin origin will be a wrong statement, because this is to indicate what? Their Latin origin, and that is the incorrect statement over here. Okay, this is first PUC, dear. Hi Shreya, this is first PUC, dear. Cool. The next question that we have, phycoerythrin and fluoridine starch are found in. Phycoerythrin and fluoridine starch. This is a unique feature of what? Red algae. What is the other name for red algae? Rhodophyceae. Please remember, how many algae is there? How many algae is there? Chlorophyceae, Phyophyceae, Rhodophyceae. Okay, there are three types of algae. Chloro green, Phyo brown, and Rhodo red. Okay, so green, brown, red. There are some unique features that you need to know about each of them. That is, all of them have chlorophyll pigment, and that's an easy way to remember. Chlorophyll A is common over here. After A comes what? B. So from here onwards, chlorophyll B is there, here chlorophyll C is there, here chlorophyll D is also going to be there. So please know that. Apart from that, if you're talking about brown algae, they have an extra thing that is known as fucoxanthin. What is it? Fucoxanthin. Whereas when you talk about red algae, for the red color, they have one more thing that is known as phycoerythrin. That was the question. Okay, phycoerythrin. So please remember. Okay, Chlorophyceae, green algae, has chlorophyll A, along with that B also. Here A along with that C. Here A along with that D. That is how it goes. Here you have an extra thing that is known as fucoxanthin for Phyophyceae. Here you have phycoerythrin for Rhodophyceae. Now, what is the storage food? That is also important. Stored food in the case of Chlorophyceae is starch. Okay, and if at all you see a term that is known as pyrenoid body. Pyrenod, that is also something that acts as a storage unit. Okay, it acts as a storage unit where P for pyrenod and P for protein. So proteins are also going to be stored in the pyrenod unit along with starch. So please understand, starch is there and pyrenod body is something that is going to store a lot of things. So please remember that as well. In Phyophyceae, it is mannitol and laminarin. Mannitol and laminarin. In Rhodophyceae, it is going to be fluoridine starch, just like how we saw over here. Fluoridine starch is what? The unit, the stored food out there. So one has just starch and pyrenod, the other one has laminarin and mannitol, and the third one has fluoridine starch. Okay, fine. Now the cell wall over here, you have plain cellulose. You have plain cellulose in Chlorophyceae. They are going to be the one that is very, very simple. This is just cellulose. This is going to be having cellulose plus a covering of algin. Plus a covering of algin. Over here, you have cellulose pectin and polysulfate. My dear children, I'm not asking you to remember everything. It's not possible. But if at all you're able to recollect anything, it will be of good, right? So that is why I'm just telling about this particular thing. Now finally, you have the flagella number and position of insertion. So basically when they make spores, so they reproduce with the help of spores. So the spores which are going to be produced, they will have flagella. How many? 2 to 8 flagella will be there. In the case of chlorophyceae, they have the highest of them, and basically all of them are going to be equal. They are going to be equal. So if this is a spore, you have 2 to 8, all of them are equal, and all of them are found at the apical position. That means at the top position in chlorophyceae. When you talk about phaeophyceae, things are going to be different. Over here, they just have 2 of them, and that is laterally arranged, and both of them are going to be unequal. That is the case of phaeophyceae. Just imagine that someone is hugging. Okay, and basically this is a unique feature. In the case of rhodophyceae, they do not have any flagella. So we say they have non-motile spores. So non-motile spores is a unique feature of what? Rhodophyceae. So that's all. Please remember, please try to remember at least some of these things which I have told you over here. Okay, now apart from that, in the case of plants, few things that you can remember, plant kingdom is that bryophytes which are there, bryophytes have liver whorls and mosses, liver whorls and mosses, and you have few things which are unique to them. For example, liver whorls have gamete cup. Gamete cup was a question the previous year which says that it is asexual, it helps in asexual mode of reproduction, and it is a multicellular body. Now the term is important. If at all you do not understand what is there, it's okay. At least try to remember the term. Liver whorls, gamete cup, and they also have something that is known as thallus. Thallus is nothing but the flat plant body out there. What about mosses? In mosses, Baba, you see something which is known as, just a second, you see something that is known as protonema. What do you call this? Protonema. This is very unique feature. The first gametophyte that is being produced is protonema. From this primary protonema, you get a secondary protonema, which is going to be the leafy stage of it. So protonema or the leafy stage is unique to what? Bryophytes, and in bryophytes it is unique to what? Mosses. If you can Okay, now pteridophytes, Baba, the most unique thing that is there is sporophytic stage is going to be dominant over there, and they have something that is known as prothallus. Prothallus. Okay, in gymnosperms, the unique thing that we have is what? Naked ovules. They have naked ovules. They do not have fruit formation. Now, in the case of angiosperms, the unique thing is that they have double fertilization. So, bryophytes, Baba, what do you have? Gamete cup, thallus, protonema, and the leafy stage. If at all you can remember, just remember. Pteridophytes, the unique thing that is there, prothallus. Okay, prothallus. Now, gymnosperms, naked ovules. That's all. We cannot do anything else. Okay. The next thing that we have is in the maize plant, CO2 fixation occurs in both mesophil. There I'll do PU2 also. Okay. In maize plant, CO2 fixation occurs in both mesophil and bundle sheath cells. The enzyme involved in these cells for the respective process is what? C4 plants. Okay. Maize plant is what? C4 plants, and this is something that has been frequently asked. Let's try our luck. Let's see if at all it's possible to get our marks over here. So in C4 plants, you have two arrangement. You have mesophil cells first, and then you have bundle sheath cells. You have okay, masters in business, just remember, mesophil first and then bundle sheath. In mesophil, first of all, the CO2 come and basically it is fixed. With the help of which enzyme? It is PEP carboxylase, PEP carboxylase in mesophil cells. Okay, now it is fixed in the form of what? It is fixed in the form of oxaloacetate. Lot of things happens over here. Finally, the CO2 which has been fixed is released back into the next cell, which is known as what? The bundle sheath cell, where again the CO2 is being fixed. By whom? By Rubisco. By what? Rubisco. Why is this arrangement found in C4 plants? Because in C4 plants, Baba, if at all the Rubisco is already available, the enzyme is available, it can go and bind with oxygen, and we do not want that to happen. That is known as photorespiration. It will lead to loss of energy. So we do not want that thing. That is why we have a different type of anatomy, which is known as Cran's anatomy, and this is found in C4 plants such as maize plant, where you see mesophil and bundle sheath cell. First PEP carboxylase, second thing is Rubisco. If you can remember, please remember. Okay, the next thing, when triopalmitin is used as a respiratory substance in aerobic respiration, the process consumes 145 molecules of oxygen, releases 102 molecules of CO2. What is RQ value? Listen, respiratory quotient is very simple. Respiration, what happens in respiration? Oxygen is being utilized, CO2 is being evolved, right? CO2 evolves and oxygen is being consumed. So the reaction, or basically the formula for that is this. Please remember the formula if at all they ask the formula. Now, apart from that, if at all they ask about what is is going to be the respiratory equation for fat, for carbohydrates, for proteins. Just remember this thing, that is going to be helpful for you. You have carb, you have fat, and you have protein. Right, now over here for carbs it is always going to be 1. This is equals to 1. Unity is what you call it. Okay, now for fat, F is like this, right? You make the ulta of F, just twist it off, it looks like 7, so it is going to be 0.7. What about protein? It is going to be 0.9. Similar to P, make ulta of it, you get 0.9. That is how you can remember it. So how do you know it's a fat or not? Basically, tripalmitin is going to be fat. So just give your answer. You do not have to do the calculation. Your answer is going to be 0.7 itself. So carb, it is 1. For fat, it is ulta of F, that is 0.7. And for protein, it is going to be ulta of P, that is 0.9. This is the basic logic that you have to apply. And basically, with the help of it, you can get marks if at all this type of question comes in. Please remember the formula. That is your part. Okay, next question that we have is, during citric acid cycle, the various organic acids undergo decarboxylation. So basically they are asking you, in TCA cycle or citric acid cycle, how many number of carbons are there? So always remember, TCA cycle begins like this. You have oxaloacetate. Oxaloacetate has 4 carbons, onto which acetyl-CoA is going to come and basically bind. Acetyl-CoA has 2 carbons, and that's how we get the first thing that is known as citrate, and that is why you call it citric acid cycle or tricarboxylic acid cycle. What is happening over here? From 4 plus 2, you got 6 carbon compound, and here you do not have to mug up things. Always remember the first 2 guys are having 6. Okay, alpha ketoglutarate is the only one that has 5 carbon, because 1 carbon is lost in the form of carbon dioxide. Then from this point onwards, every one is going to be 4. Succinyl-CoA, succinate, fumarate, malate, oxaloacetate, everything is going to be 4. So 2 guys are going to be 6 carbon, that is citrate and isocitrate. 1 guy is going to be having 5, that is alpha ketoglutarate. This is the unique one. This is the only one that has 5. Rest everyone, Baba, has 4 carbon only, nothing else. So basically you have been asked which has 4 carbon, 5 carbon, 6 carbon. We know that there is only one that is having 5 carbon, and that is alpha ketoglutarate acid. So if at all that is in the middle, that becomes your answer. This is in the middle, and this becomes your answer for this particular question. Succinic acid is nothing but succinate only. They have 4. Citric acid and isocitrate are the only ones that have 6. So that's all. Okay, second PUC, I'll be doing it right now. Don't worry. The next thing, identify the incorrect statement. Kynetin is a derivative of adenine, which is pyrimidine. So kynetin is a derivative of adenine, but that is a purine, not a pyrimidine. The technique of decapitation is widely used in tea plantation. What happens, Baba? Basically, you have auxin, and auxin is there at the tip of the plant. When auxin is there at the tip of the plant, it makes the plant grow like this, not like this. It makes the plant grow like this. If at all you cut off the tip, what is going to happen? Now it is going to give lateral branches, and that is what the tea plantation people want. They cut it off. They do decapitation. Why? Because auxin brings apical dominance. If that is removed, you have lateral branches. Ethylene is a gaseous plant hormone. Yes. GA3, gibberellin, basically hastens the malting process. Always remember, even if you do not understand the meaning of malting, GA3, gibberellin, is basically responsible for 2 things. One is malting and other one is bolting. Malting and bolting. Malting basically means conversion of starch into simple sugars, and basically bolting means what? Bolting means internodal elongation. It makes the plant grow really fast such that it can do flowering, and this is found in the case of roseate plants, such as cabbage and stuff like that. Okay. Cool. So ABA is a growth promoter. Wrong. ABA is going to be a growth inhibitor. It is going to be a growth inhibitor. So this is wrong because it is a purine. Adenine is a purine, and over here this is a growth inhibitor. Going ahead, I told you auxin is for apical dominance. Gibberellin, which is there, is for bolting. And now going ahead, you have diagram given here in the standard ECG of a normal person. So ECG over here, you have P, Q, R, S, and T. Listen, first thing that happens in our heart is what? Contraction of atrium. Atrium first, and then the ventricles, right? So contraction of atrium happens. Blood is given to what? The ventricles. This contraction of atrium is denoted by P. Okay. Now blood is going to the ventricles. The ventricles have to contract. This contraction of ventricles is being denoted by what? QRS. Now the ventricles which have contracted, they have to relax, and that relaxation basically is being denoted by what? T. Okay. So atrial relaxation is not being mentioned over here. Atrial contraction, ventricular contraction, and ventricular relaxation is going to be mentioned over here. Always remember that contraction happens because of what? Depolarization. Depolarization, and relaxation happens because of what? Repolarization. If at all you're finding it difficult to remember, R for relaxation, R for repolarization. Always remember. So depolarization of atria, both of the atria, depolarization of both of the ventricles, and repolarization of both of the ventricles. This is how things are going to go. Okay, this is how things are going to go. This is PQRST. So contraction of both atria, contraction of both ventricles, and relaxation of the ventricles. You're not talking about relaxation of the atria over here. Contraction happens because of depolarization in both of these cases. Relaxation happens because of repolarization. R for relaxation, R for repolarization. I hope this is okay. Okay. Now, one more very important thing, tissue patch found in the left upper side. Okay, so this is about the SA node and things like that. Just listen to me carefully. You have your atrium and the ventricle over here, right? Atrium and the ventricle. Always remember, if this is the atrium, the right atrium, this is the left atrium, this is the right ventricle, and this is going to be the left ventricle, right? Here at the top right side, not the left side, you will have what? SA node, sinoatrial node. It sends signals to all the muscles and causes contraction of the atrium. What do you have? SA node towards the right side of the right atrium. Now towards the downside, what do you have? Over here in between the atrium and the ventricle, you have AV node, but this is towards the left side. This is towards the left side. What do you have? Atrioventricular node. This sends signal over here. This is known as bundle of His, and bundle of His has certain fibers which is known as Purkinje fibers. It is known as Purkinje fibers, which is going to contract things. Done. Okay, so that is how things are going to go. And always remember, SA node is auto-excitable. You do not need anything else. It do not need any external stimuli. It can work by its own. So always remember that this is towards the right side, this is towards somewhere in the middle, and mostly towards the left side, that is AV node. Cool. Now refer to the given diagram and select the correct option, PQRS. So always remember, I cannot explain all of it as of now, but remember, oxygen which is there, it is found in the most amount in our Earth's crust. So if at all you find oxygen in the most amount, that is 46.6, that would be your answer. What about sulfur? Sulfur, Baba, over here it is going to be 0.3. Sulfur as well as carbon dioxide. Okay, so not carbon dioxide, carbon. Sulfur and carbon which is there, both of them are found in equal amount, that is 0.03 in the Earth's crust. It is going to be more in the case of our body, but case of sulfur, it is going to be 0.3 in the body as well. So in the body and the crust, it is going to be less only. In the Earth's crust, it is going to be even lesser than the body, and that's the only thing. So oxygen, the highest. Okay, oxygen is going to be the highest. Sulfur and carbon dioxide is going to be the lowest in the Earth's crust, and in the human body, if at all you talk about that, sulfur is still low. It is just 0.3. Cool? Yes. Fine. Now going ahead, refer to the given. So the only thing that you have to remember is, if at all you get a question like this about this particular thing, GLUT4 is nothing but some important transporter which helps glucose to get inside the cell. Okay, that is GLUT4. Trypsin is an enzyme. It's a protein, but it's an enzyme. Insulin is also a protein, but it is a hormone. It's not an enzyme, it's a hormone. Antibody, you know, it's fight against infectious agents. Collagen is something that is found in between the cells. It is the intercellular ground substance out there. It is what? Intercellular ground substance out there. If at all you get questions like this, make sure you get it. So over here the question is, X is a type of protein that act as hormone. So hormone is what? Insulin. Dash is a type of protein which enables glucose transport. It is going to be GLUT4. So these type of questions could be coming in. Make sure you check it out. Okay. Now read the given statement and select the correct option. This is regarding what? The prophase and that is meiosis I. Listen, in meiosis I you have prophase I and metaphase I, things like that goes on, right? So in prophase I of meiosis, you have leptotene, zygote, picotin, diplotin and dikinesis. The easiest way to remember is it. Lazy zebras play during day. Lazy zebra play during day. And you just have to remember the most important part of it. In the leptotene, what is going to happen? You have chromatin fibers. Chromatin fibers, Baba, they are going to condense. They are going to condense to what? Chromosomes. They will look like chromosomes. So thin thread condensation happens in leptotene. In zygote, for the first time, Baba, chromosome. Okay, chromosome from the father and the mother. Basically, they have duplicated themselves during the S phase, right? Now, they both will be coming together. They are going to come together and they are going to form synapses. This is a unique feature. Synapses is going to be formed. Why? Because over here, you have synaptodermal complex being formed, and this structure where you have these things is known as bivalent. It is known as bivalent, and basically, Baba, if at all you see, this is actually one particular chromosome, and these two things are what? Sister chromatids. So it got duplicated, that is why sister chromatids. Here also sister chromatids. But when you talk about this one and this one, they are going to be non-sister chromatids. Okay, they are two separate chromosomes, and this thing is going to be known as non-sister chromatids. Anyways, the thing that is being formed over here is known as synapses, and basically it is known as bivalent. How many chromosomes or basically chromatids do you have? You say it's a tetrad that is being formed. That means 4 of them are going to be coming together. 2 chromosomes are only there, but they have two branches or chromatids. That is why we call it tetrad. Always remember that. So that is the feature about zygote. Now since they're very close, Baba, what is going to happen? When you're very close, you might be exchanging some information, right? So that is exactly that is going to happen over here. In this particular case, Baba, crossing over is going to happen, and this is the most important thing. When they cross over, you need an enzyme. What is the name of the enzyme? Most important thing, the enzyme's name is recombinase. It is known as what? Recombinase. It is known as recombinase. Please do not forget it. Recombinase helps in the crossing over process. That happens in pichitin. Now in diplotin, what is going to happen? They are going to break up. When they break up, when you break up, obviously you might be still attached, right? At some point you might be still attached. These attachment time. So over here until now they were closed. Why? Because they had a glue in between. Now the glue has went off. When in the diplotin stage, when the breakup is happening, the glue has went off, but still they are attached at certain points. You call those points as what? Chiasmata. What do you call this, Baba? You call it as chiasmata. Okay, now chiasmata is there. They are still attached to at certain point. Finally, dikinesis, they are going to die. That means the last point, terminal stage is there. So we say terminalization of chiasmata. Finally, when you move them apart, what is going to happen? The chiasmata, suppose it was here, it will move upwards because you are tearing it apart. You got it? So terminalization happens over here, and also the nuclear membrane breakdown, because that is a feature of prophase. The nuclear membrane also breakdown. Please remember the unique feature about each of them. Lazy zebra play during day. Lazy zebra play during day. That's the only thing that you can remember it. Okay. Yes. Okay. Now over here, talking about glycolysis, GGFFDG That is how it is going to go. So glucose, from glucose you get glucose 6-phosphate. Then you make it to fructose 6-phosphate. From fructose you make fructose 1,6-bisphosphate. What is happening over here? I am adding a phosphate group. That is why glucose 6-phosphate. Now you got fructose 6-phosphate. Again I am adding a phosphate group. So in both these places ATP are going to be utilized. So GGFF. Until this I hope it's clear. From fructose 1,6-bisphosphate is a 6-carbon compound. You're going to break it down into 3,3-carbon compound. One is going to be dihydroxyacetone phosphate, and the other one is going to be glyceraldehyde 3-phosphate. If at all you are getting the option to arrange them, at least you can arrange them in this particular order. Okay, so you have both of them being formed by this particular guy only. This particular guy forms both of them. They are both 3,3-carbon compound. What is going to happen? You do not need DHAP, so you convert it into glyceraldehyde. So basically I have 2 molecules of glyceraldehyde 3-phosphate right now, and this 2 molecules of glyceraldehyde 3-phosphate forms what? 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate out there. Again, over here it is losing 1 phosphate, so it becomes 3-phosphoglycerate. Again loses 2-phosphoglycerate. Okay, it becomes what? 2-phosphoglycerate. And finally you get phosphoenol pyruvate, and then you get pyruvate. Very, very, very important thing. So this is the flow. If at all you can just get it. Okay, ma'am is getting revised, not us. Is it? Is it too much? Is it helping someone at least? Guys, is it helping you recall anything? Yes, once more, I can do that. Listen to me very carefully. And yes, it is helping. Okay, listen. So basically, what do you have? You have glucose and... Okay, just give me a second. Okay, just give me a second. Understand it in this speed. Yeah. Okay, so guys, for those of you who just want the PDF, I have tried my best. I have spoke. Okay, I have spoke about it. They will be soon putting the PDF, is what I hope so. in the channel. Okay, let's go. So basically, I'll just revise once more. Glucose is there, you get glucose 6-phosphate. Listen, with this particular picture, it will be more easy. Glucose is there, I got glucose 6-phosphate, I have added up one phosphate group over here. Now what do? You convert it into fructose 6-phosphate. Fructose 6-phosphate, which is there, I am adding one more phosphate group. That is why it becomes fructose 1,6-bisphosphate. Okay, 1,6-bisphosphate. Bisphosphate means it has two phosphate group now. In 6-carbon compound, at 2 position, the first position and 6th position, it has phosphate. Now this particular thing divides into two things, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Though both of them are 3-carbon compound. Both of them are going to be 3-carbon compound. But I just need this, not this guy. So I'll convert it into glyceraldehyde 3-phosphate. That means now I have 2 molecules of glyceraldehyde 3-phosphate. Got it? This glyceraldehyde 3-phosphate converts into 1,3-bisphosphoglycerate. What? It is 1,3-bisphosphoglycerate. From 1,3-bisphosphoglycerate, what are you going to get? You are going to get 3-phosphoglycerate. And now you're going to make the numbers less. 1,3-bisphospho, then 3-phospho, then 2-phosphoglycerate, and finally phosphoenol pyruvate. And all of that two molecules are being formed. Phosphoenol pyruvate finally forms the pyruvate molecule. Just 2 molecules will be formed. But pyruvate has 3 carbons, so 2 molecules of pyruvate is being formed over here in this particular case. That is known as glycolysis. It is known as glycolysis. If at all you need to remember this, remember GG, FF, DG, B, and then 4P. And then 4P, if you can. I'm telling you, it's not easy for everything to be remembered, but yes, still you can try. Now, epigynous flower is what, Baba? Epigynous means the ovary over here, Baba, it is going to be inferior. If at all you look, you have 3 conditions. Hypogynous means it is going to be superior at the top, and everything would be below it. Petals, sepals, every part of it will be below it. Perigynous means all of them are going to be somewhere in the same level. Epigynous means ovary is going to be inferior, everything is going to be at the top of it. It is known as epigynous condition. Okay, it will be known as what? Epigynous condition. Now over here in the case of glycolysis, someone is asking how many ATPs are being utilized. 4 ATPs which are there, it is being formed. 2 ATPs are being utilized, which is why 4 minus 2, at the end you get only 2 ATP. At the end only 2 ATPs are being formed. Okay. Yes. Now the next thing that is there is identify the floral formula of plant belonging to the potato family. The potato family, Baba, is coming under Solanaceae. Always remember, it is going to look like this. You have the female and the male part right over here, right? That is going to the first direction. Now the next thing is that how many sepals are there? Sepals, the green ones. Together you call them as what? Calyx. What do you call it? Calyx. You get 5 of them, 1, 2, 3, 4, 5. So you put 5. And now if at all you see, they are all fused together. So I put a bracket in between. Similarly, talking about the next thing, that is what? The petals. Petals are how many? 5 of them. So how do I remember petals? Petals means corolla. Group of petals is corolla, and there are 5 of them. Again, Baba, if at all you see this particular picture, they are also fused amongst each other. So I'll put it that also in bracket. Now next is what? Next is androgen. That is the male part. How many of them are there? If at all you see the stamen, 1, 2, 3, 4, 5. 5 of them are there. But are they fused with each other? No, Baba, they are not fused with each other. So I'll write 5, but they are not fused. Okay, ma'am, but I see that they are fused with the petals. They are attached to the petals, isn't it? This is the petal and they are attached to it. So what do? I make a connection like this, because petals means this only. Okay, so I make a connection like this, and that is known as epipetalous condition. Epipetalous condition. And finally, what do you have, Baba? You have gynoecium. Gynoecium, how many? You have 2 of them, and 2 of them basically are attached to each other. Gynoecium, 2 of them are attached to each other. So I'll say that you have gynoecium and you have 2, and basically they are fused with each other. And always remember, it's a superior ovary. That is why you denote it as G and you put a dash below it. That means it's superior. Superior means it will be in the middle. Only superior cases is going to come in your examination. No, nothing else. Okay, cool. Now cells in the quiescent stage, cells which are there in G0 stage, quiescent stage, they are not actively dividing, but you need to understand they are metabolically active. Inside the cell, things are ongoing. They are not dividing, but they are metabolically active. Please remember that. Okay, now bulliform cells are the characteristic feature of what? Bulliform cells are found in the case of grass leaf. Please understand, monocot, grass leaf. Thus remember this, bulliform cells, grass leaf. Bulliform cells, grass leaf. That's all. And if at all, guys, you want just the PDF, as I told, you can just join the WhatsApp channel and get the PDF. Okay, that's the least thing that I could do for you if you just want the PDF. If you want me to tell things, please be here. Now we are starting the second PUC portion. Listen to me carefully. If at all you get a condition like this, how many gametes are being formed? You use the formula 2 raised to the power n. N is what? Number of heterozygotes. Baba, how many heterozygotes are there? This is a heterozygote, this is a heterozygote. This is not, because both the alleles are similar. So if at all you see, 2 of them are heterozygotes. So I put 2 is raised to the power 2. It becomes what? 4. So the answer for this question is going to be 4. This formula is important. N is number of heterozygotes. Heterozygotes means when the alleles are not same, you call them as heterozygotes. So this is 1, this is second. Rest is A, both the alleles are same. I will not count it as heterozygote. Okay. Now the next thing which is there is select the incorrectly matched pair. Listen to me. Pleiotropy, one of the most important thing. Pleiotropy means one gene, multiple effects. One gene, multiple effects. The best example for that is phenylketonuria. One enzyme, phenylalanine hydroxylase, is not present. That is why you get 2 effects being seen. One effect is that phenylalanine which is there, it is going to accumulate in our body in the form of phenylpyruvic acid, and that causes mental retardation. Second effect is seen that this, since conversion is not happening, tyrosine is a very important thing for the formation of melanin, and melanin is important for pigmentation. So our pigmentation is also not going to be happen. We find hypopigmentation. Our hair and our skin will be finding what? Hypopigmentation. Always remember. So one gene being affected, multiple characters being affected. This is known as pleiotropy, not polygyny. Do not get confused. Height in human beings is polygyny's example. That is ulta of this. Ulta means what? If this one gene is affecting multiple traits, in that multiple genes are there. Multiple genes are there, and these genes are affecting one trait. That is polygyny. The name itself is understood. Multiple genes, polygyny. Height and your skin color is an example of polygyny. If at all you have the least combination like this, all of them are recessive. It will be the lightest skin tone. If at all of them are going to be capital, it is going to be the darkest skin tone. Always remember. Linkage is found in Drosophila. Yes, linkage is found in Drosophila. Always remember, if at all 2 genes are linked very close to each other, they will find less recombination. That means you and your friend are very close to each other. You will not allow someone else to come in your friendship. That means no new recombination. Always remember that. So in that, yellow body and white eye. Okay, yellow body and white eye color genes are present on the chromosomes. Both of them are very close to each other. They will show very less combination. Always remember, yellow body and white eye. Just imagine 1.3% recombination is what? Very less recombination. Why? Because they are tightly linked. Whereas over here you have one more gene present at the tip, that is known as miniature wing gene. Over here, when you talk about the linkage, how they are loosely linked. They are far away from each other. If they are far away from each other, don't you think some other friendship can also happen? Yes, new combination can happen. So we say the percentage of recombination is 37.2%. What is it? It is 37.2%. The maximum amount of recombination is 50. Here it is 37.2% recombination out there. One more thing that you have to remember is linkage and recombination was given by Thomas Hunt Morgan. Thomas Hunt Morgan. Now his student's name was Alfred Sturtevant, and he said that if you give me the percentage of recombination, basically I get to know the distance between the genes and I can locate the gene. So he was the person who gave gene mapping. So you are my students, you gave what? Gene mapping. I am the teacher, I gave what? Linkage and recombination. That's all. Okay. Now incomplete dominance which is there is found in snapdragon or antirrhinum species. Incomplete dominance means one dominant allele is not completely dominant over the other one. You have seen in the case of snapdragon, the red and the white one gives pink flowers, right? Now codominance is seen in ABO blood group. That is absolutely correct. A and B, both the alleles are equally dominant. That is codominant. Okay. Now the next thing that we have is this. In the following pedigree chart, the mutant trait is shaded black. The gene responsible for the trait is what? So guys, over here, before I go ahead, I want to ask you one question. Please answer me. Until what time can you see the live? Until what time can you see this particular live? Can you tell me the time? Because I know you have to enter the examination hall. Can you please tell me the time when you have to enter the examination hall? 1:15. Oh my God. Okay. Cool. So listen to me carefully. Over here you have the parents and you have the progeny. If at all you see skipping of generation, that means here the parents are not affected but the children are. Here the parents are not affected but the children are. So we see that it is recessive disorder. If at all you find skipping of generation, I'm talking about this generation, then this is the next generation out there. If at all you see skipping, it is going to be recessive. Now always remember, there is very less chance for them to give what sex-linked disorders. They are usually going to give autosomal because that is there in the NCERT. of the pedigree chart I'm talking about. Okay, so over here it is autosomal. Why? Because the male and the female is somewhere equally affected. If at all it is sex linked, you will find crisscross pattern. You will see one male is affected. If the male is affected, their daughter is going to be affected. If the daughter is affected, their son is going to be affected. That is crisscross. Okay, from male, female, and then male. That is how it is going to go. Otherwise, it is to be female, male, and then female. That is how it is going to go. That is crisscross pattern. This is not that. This is recessive disorder. Why? Because it is skipping generation, and autosomal because both the sexes are equally affected. Now, if at all you get a if at all you get a picture like this. So this was for autosomal recessive. How many autosomal recessive disorders do you have? Sickle cell anemia, phenylketonuria, thalassemia. 3 of them are autosomal recessive disorders. Always remember. Also cystic fibrosis, not given as a thing to understand, but that is also there. Cystic fibrosis is also a recessive disorder. Please know that. Okay. Now talking about this particular thing, that is myotonic dystrophy, only autosomal dominant disorder that is given in your NCERT, the picture. How do you understand dominant? Every single generation has one person being affected. Here mother, here the son, here again the mother of this particular guy. So always in every single generation one person affected, dominant disorder. Okay, if at all that is sex linked, you would have got to know, but here all the females and males are almost equally affected, so it is going to be autosomal. So autosomal dominant disorder, only one example is there, and that is what? Myotonic dystrophy. Myotonic dystrophy. Okay, and you have sex linked recessive disorders only, and there are 2 of them, color blindness and hemophilia. That's all. Now going ahead with the next thing that is there, which of the following are true about male honeybees? If at all you talk about honeybee, just understand the logic over here. You have the female honeybee queen. Okay, she has 32 number of chromosomes, whereas when you talk about male honeybee, they have half of it, that is 16. Now if someone has 32, they can undergo meiosis. When they want to make gametes, they will undergo meiosis and they will make gamete that has 16 number of chromosomes. But if this guy already have only 16, how can it undergo meiosis? So it undergoes mitosis. What does it undergo? Mitosis. It undergoes mitosis to produce what? 16 number of chromosomes over here. Okay, no meiosis. Now listen to me. The female egg that is over there having 16 number of chromosome, the male sperm having 16 number of chromosome, when both of them come together, what do you get? 32 number of chromosome. It is going to be a female or a male? Female. Why 32? Because 32 is found only in the case of females. Diploid is found only in the case of males. Sorry, females. Then how do you males form? So males are formed without fertilization. No fertilization is going to happen. Without fertilization, you get the males, and males are going to be haploid. This process is known as parthenogenesis. And because of this, always remember, the guy doesn't have a father. The guy does not have a father, because it is found without fertilization. But does it have a grandfather? Yes, because the mother was formed because there was a father. So it has a grandfather but no father. Please remember it. This is also an important thing. Okay, so only 1 and 2 are the correct things about thing. One is what? They have 16 chromosome per cell about male bees, and the third one, they can have grandfather but no father. This is a question. This is not something that I made up. Okay, now a dihybrid cross between a true breeding round yellow seeded and true breeding wrinkled and green. Whenever you get that dihybrid cross and they are asking you for the segregation of anything, please understand, use this logic. Okay, what do you have to do? You have to use this particular logic. 9:3:3:1. 9 of them are always going to be the dominant character. What is a dominant character, Baba? It is going to be round and yellow. It is going to be round and yellow. What about the recessive character? It is always going to be wrinkled and green. Wrinkled and green. The recessive character, recessive parent. Remember? Now, 3 and 3 of them are going to be non-parental, something that is unique, something that is new. Over here, round was always with yellow. So what do? We make this time round with green. Round with green. Okay. Wrinkled was always with green. This time we make wrinkled with what? Yellow. This is the non-parental character. What is the question? The ratio of segregation of round and wrinkled. They are not asking you how many of them are round and green and things like that. They are asking you round and wrinkled. So you use the logic. 9 plus 3. 9 plus 3 of them are round. That means 12 of them are going to be round. Okay. 3 plus 1 are going to be wrinkled. 3 plus 1 are going to be wrinkled. So what is the ratio over here? 12:4. That means what? 3:1. The same ratio that we got in the monohybrid cross. Similarly, they can ask you about green and yellow. The same logic is going to be applied over here. The yellow ones basically 9 plus 3, 12, and the green ones is the recessive character. So it is going to be 4 anyways. So please know the logic. It is 3:1. That is going to be the answer for this particular question. I hope that works. Yeah. Now the next question that is there, a pure breeding pea plant with round yellow seeds are crossed with pea plant having wrinkled green seeds. On selfing of F1 of this hybrid, 64 progenies were obtained. The same question. Again, they are asking you the same question. That means basically the same thing is happening, but they are clearly mentioning about one thing. That is what? You get 64 progenies. How many progenies did you get over here? 9 plus plus 3 plus 3 plus 1 is equals to 16. You get actually 16. But now the scenario has changed. They are saying 64 of them are there, and they are asking you how many of them are going to show non-parental character. How do you find it out? It's very simple. Out of this, I told you 3 are the non-parental one, right? So out of 16, Baba, 3 plus 3, 6 of them are going to show non-parental, and you have to find like this for 64. So what do? You have formula 6 by 16 into 64. Now if at all you do this, you get 24. 6 by 16, out of 16, 6 of them are non-parental. Now you do the same for 64 of them. So you do the calculation, you get 24. You will get what? 24. Okay. So that is the logic. So the answer for that question is 24. If at all you have been asked about parental, then 9 plus 1 is equals to 10. So 10 out of 16 into 64 will give me what? The parental one. So same logic applies over here. I hope this is clear. Now, which of the following statement is being correct? Change in the whole set of chromosome is called aneuploidy. No. Change in the whole set of chromosome is known as polyploidy. Aneuploidy means you have lost one chromosome or you have gained a chromosome. It's not multiple. It's not whole set of chromosome. Whole means it has to be polyploidy, and polyploidy is seen in the case of plants. P for polyploidy and P for plants. Now sickle cell anemia is a quantitative problem. Wrong. It's a qualitative problem. What qualitative problem is that? Basically, beta globin chain of hemoglobin is being affected. In the 6th position, Baba, there is a change. There is a change. Okay, it was supposed to be GAG, but now it is GUG. It was supposed to be GAG, but now it is GUG. A comes before U, so always remember it was this, but it became this. So basically, this was coding for glutamic acid, but now it is coding for valine. Again, G comes before V, so G was like this, then it became this. Now valine is there, the whole beta globin chain is going to change up, and that is going to mess up with the quality of the hemoglobin. So it's not a quantitative, it's a qualitative problem. Now thalassemia is a qualitative problem. Wrong. Thalassemia is a quantitative. Quanti has a T, and T for thalassemia. Always remember that. Qualitative also has, but quantitative has more T's. Okay. Now the next thing, female carrier for hemophilia may transmit the disease to the sons. Is that particular thing correct? Female carrier for hemophilia. A female carrier means she has one chromosome being affected. Now can she pass the disease to the son? Yes. To the son she can only pass the X chromosome because she has only that, right? Now she can pass this or she can pass this. She can pass the one that is problematic. Yes, it can be done. Okay, it can be done. So this is the only one that is correct. Now XO type of sex determination and XY type of sex determination are the examples of XO. Over here, if at all you see in the grasshopper, Baba, in the case of insects, the female are going to be XX, the male are going to be XO. XO means their sperms can carry either X chromosome or their sperms will carry no chromosome. Either way, if at all you see, they are producing two type of gametes. This is known as heterogamety, and when males are doing it, it is known as male heterogamety. This is found in us also, in our human beings also. XY, X type of chromosomes, or basically Y type of chromosomes are being sent through males. So again, male heterogamety. Where do you see female heterogamety? Female heterogamety is seen only in the case of birds, where they have ZW, W for women, always remember, and ZZ for males. So they are producing the same type of gametes. But what about the females? Either Z or either W. That is how things are going to go. Either Z or W. Got it? So the example for female heterogamety is going to be birds. For the example for male heterogamety, it can be insects. It can be XY is found in the case of human beings and Drosophila. XY both in the case of us and also Drosophila. For birds, it is ZW. Always remember that. Okay, now a man with blood group A. Okay, a man with blood group A marries a woman having blood group B. When I say I have A blood group, please always remember, it can be something like this, where both the alleles are same, or it can be something like this. It can be both the cases. Similarly, when I say marries a person with B blood group, so B blood group can again be like this, or it can be also like this. Why? Because B is dominant over O. Similarly, A is dominant over O. It will give A blood group, it will give B blood group. Now, you have been asked about the maximum possible. So which one will you consider? You will not consider this cross. You will consider the cross between both of them for finding the maximum ones. So what do you do? IAIO, and I have IB, IO. When you cross this and this, you get IA, IB. A and B, both of them come together. That means both of them are equally dominant AB blood group. When this this and this comes together, I get IAIO. A is dominant over O, I get A blood group. When this and this comes together, I get IBIO. B is dominant over O, I get B blood group. When this and this comes together, I get IOIO. That means O blood group. Now you just imagine A blood group person marrying B blood group person and still having the children with AB, A, B and O. These are the maximum number of genotypes, or basically maximum number of phenotypes that can be formed over here. Genotypes mean genetic makeup, phenotypes means physical appearance. Okay, phenotype means physical appearance. If at all you have been asked about phenotypes and you have, for example, two of them are giving A, do not count it twice, count it only once. Okay, guys, those of you who have to go to the center, wishing you all the best. Make sure you give your best. Okay, now both male and female have normal vision, though their fathers were colorblind and mothers did not have any gene for colorblindness. The probability of their daughter becoming colorblind is what? Both male and female have normal vision. When I say male is having normal vision, he has to be like this. Okay, there is no option. But when I say a female is having normal vision, she can be like this or she can be like this also. She can be a carrier. Always remember that. How do we understand whether she is this or this? We talk about the parents. So both male and female have normal vision, though their fathers were colorblind and mothers did not have the gene. So father of this particular lady was colorblind. So in that particular case, if he was colorblind, Baba, he has to be like this, and he will definitely pass this particular chromosome, because to females he can just pass the X chromosome. So obviously the daughter which is there will definitely be something like this. She will be a carrier. She will have normal vision, but she will be a carrier. Now you have to cross between this and this. That is the question. The probability of their daughter becoming colorblind. You have to find the probability of them being colorblind. So how do you do that? Let's cross and let's see it. So just give me a second. Okay. So over here, always go check the parents. Check the parents. That is definitely going to help you out. If it all is said that the female is there and the father is colorblind, the female will definitely have at least the gene. She will be a carrier. If mother and father, both of them are colorblind, then she will be also colorblind. So anyways, when you cross this with this, I get XX. She is going to be a normal female. When you cross this with this, I get XX, XC. What is this? This is also a normal female, but she is a carrier. Okay, normal vision, but with she is a carrier. Okay. Anyways, this and this, I get XY. Finally, this and this, I get XCY. What conclusion do we come with? The probability of their daughter becoming colorblind is what? 0. One is a carrier, one is having normal vision, but they are not colorblind either way, so it was going to be 0. If at all you have been asked about the son, okay, what is the probability of their son being colorblind? If at all you see, Baba, over here, 50%, 2 sons are being formed, and out of that, 1 person is affected. So we say 50% of the son is going to be colorblind. 50%. If we're talking about all their children, then we'll talk about full ratio. Then in that case, 25% of their child can be colorblind. 25% of their children can be colorblind. But when you're talking about the sons, you have to talk just about the sons. So out of 2, 1 guy is colorblind. So 50% of the boys are going to be colorblind. Always remember that. Okay? Yes. Now the next thing, which of the following statements are correct with reference to prokaryotic genome? Prokaryotic genome, Baba, are they monocystonic? No. Prokaryotes are polycystronic. Prokaryotes, polycystron. PP. Always remember. Polycystron means one structural gene has multiple genes. For example, lac-operon, ZYA. Multiple genes are there stacked up together. They form multiple proteins. That is found in the case of prokaryotes. In the case of eukaryotes, you have monocystrons. Eukaryotes means what? Us, plants, all of us are what? Monocystonic in nature. We are what? Monocystonic in nature. Okay. The next one, introns absent in structural genes. Baba, in the case of prokaryotes, they do not have introns. Always remember, prokaryotes do not have introns. I'll tell you one very important thing. In prokaryotes, there is no nucleus, nothing. So transcription, translation, everything happens in the same region. We say it's coupled reaction. Okay, whereas in the case of eukaryotes, transcription happens in the nucleus, translation happens in what? In the cytoplasm. So things are going to be different. Similarly, when you talk about introns, when you talk about eukaryotes, we have what? In the DNA, we have non-coding regions which are known as introns, and we have coding regions which are known as exons. The same thing is going to be transcribed in the hnRNA. hnRNA, you get introns as well as exons. Now, when you form the mRNA, mRNA is going to code for what? Proteins. So it will say that I need only the segment that have proteins. Please remove introns and give it to me. So now you are removing introns, and now you just have exons. You will be just having the exon, and this process is known as splicing. So removal of introns is known as splicing, and this is unique feature of eukaryotes, because eukaryotes have introns. Prokaryotes do not have introns. introns. So introns are absent in structural genes. Yes, this is absent in structural genes about prokaryotes. Transcription and translation are coupled processes. Yes, it is coupled. Primary transcript undergoes splicing. Why splicing? If at all introns are not there, what are you going to cut? Only one RNA polymerase is there. Yes, always remember, in the case of prokaryotes, they just have only one RNA polymerase. In the case of eukaryotes, we have 3 RNA polymerases. Okay, we will be having 3 RNA polymerases. That's all. Okay, now over here you have the genetic code questions. Listen, I cannot explain to you as a whole about everything. Just have the basic funda remembered. That is, you have AUG. AUG codes for methionine. It's a start codon. Okay, and this guy only has one codon. In entire methionine has only one codon. Similarly, you have one more thing that has only one codon, and that is tryptophan. UGG for tryptophan. UGG for tryptophan. UGE. Tryptophan. UG undergraduate. You can just remember like that. Okay. Tryptophan, and you have AUG methionine. What about the stop codons? You have 3 of them: UGA, UAG, and UA, UGA, and UAG. So UA, UGA, UAG, and UGA. Everything is a combination of A and G, and one is a combination of AA, UNA. That's all. Okay. Yes. Got it? Yes, mention 3 RNA polymerase. We're coming to that question here. So I'll be talking about that. If at all you just want directly the question about that. Do I have it? No. Okay, anyways, I'll just mention the 3 RNA polymerase which is there. I think that will give you some peace. Yes. So the 3 RNA polymerase which is there is RNA pol 1. Okay, RNA pol 2, RNA pol 3. Always remember, RNA pol, RNA. So first priority is given to the one that starts with R, and that is what? rRNA. It codes for rRNA, but specific types of it, everything ends with 8. So 28S, 18S, and 5.8S. Everything ends with 888. Okay, second priority is given to what? Formation of hnRNA. You just studied hnRNA right now. From DNA you get hnRNA, right? hnRNA is the precursor of mRNA. Later it forms the mRNA. That is why I call precursor. Okay, hnRNA stands for heterogeneous nuclear RNA. Now RNA polymerase 3 sounds like T, so it is the one that transcribes tRNA. It is the one that transcribes tRNA, but there are exceptions. Okay, apart from it, it also transcribes small nuclear RNA, snRNA. Apart from that, it also transcribes rRNA. But ma'am, you said rRNA is coded by this. Yes, but everyone that ends with 8, there is one that does not end with 8, and that is 5sRNA. 5sRNA is coded by the third one only. These 3 are the ones that are there. Please remember it. Try to remember it. Okay. Okay, now lacopron, Baba, lacopron, you have 3 genes. You have been asked what is what. So first one, always remember BPT, Bachelor of Physiotherapy, or whatever you want to remember B, after that comes P, after that comes T, generally also in alphabetic order. So beta galactosidase, then you have permease, and then you have transacetylase. Beta galactosidase is going to break down lactose into what? Glucose and galactose. What does permease do? Increases the permeability. What does transacetylase do? That is the last enzyme. It makes sure that everything that happens in the cell is being coordinated. That means any toxic substance buildup is not being allowed with the help of transacetylase. Okay, so over here you have the first one, that is the mRNA, and that is coding for what? Repressor protein. There is a repressor protein being coded there. Now the repressor protein, if goes and binds on the operator, transcription is not going to happen. If at all it stays here and binds with this, what is this? Inducer. Inducer can be lactose or allolactose. It can be lactose or allolactose. So this is the repressor, this is the inducer. Always remember, and these are the enzymes out there. The inducer can be lactose or allolactose. Now this is the question that I need you to do. Okay, very important thing. If at all it comes. 11th year, I have completed the 11th revision. Please check it out. The first part of it, you can just go back and check it out. E. coli was grown in the culture medium supplemented with N15NH4Cl, and total bacterial label was, the medium was changed containing N14 medium. And anyways, so basically they are asking you about this particular question, where they are saying, Baba, first of all, know the logic. You have the DNA. When the DNA replicates, what does it do? The DNA separates out and a new strand is being formed. A new strand is being formed, right? Right. Okay. Now listen to me. That is known as semi-conservative. One strand is kept there and new strand is being formed. Suppose you are growing it in an N15 medium. If at all you are growing the E. coli in N15 medium, obviously it will take up, in the DNA it will take up the N15 isotope, because nitrogen is a part of the DNA, right? So the new strands will be made up of N15. So that is the logic. So what we have done as of now is that, basically, first thing that we did is, we grew it in a medium that was containing N15 only, which is why both the strands of the DNA has N15, N15. Okay. Now we centrifuged it in what? Calcium. In what? Cesium chloride, CSCl, okay, cesium chloride. What happened when we centrifuged it? We found a heavy band. Why? Because N15 is a heavy isotope. So we found a heavy band. Okay, very important. Now listen to me. We will change the medium now. We will grow the E. coli in N14 medium now, and every 20 minutes it is going to divide. So first 20 minutes, Baba, you did it. What happened? The DNA which was there, it was segregated. It separated. The new strand you got, but this time the new strand is what? N14, right? That is why it is green in color. Got it? Okay. Now when you centrifuge it, will you find the heavy band? No, you do not have heavy band. Rather, you have a hybrid band somewhere in the middle, because N14 is light chain and N15 is what? The heavy chain, which is why you find the middle band. That is the hybrid band. Now what, Baba? Again for 20 minutes we are going to do. So that is why it is mentioned as 40. 20 plus 20, 40. Same medium, N14 only. Now in N14, whenever you do, whatever you do, it is going to take up the green one only, N14 only. So what is going to happen? And the red one cannot go, because anyway it is going to be conserved. So always understand, in whatever generation you find, you will be finding hybrid bands for sure. This is going to be there. Whatever you do, you will find the red one with the green one, because that is there in the parent. Okay, and only two will be there. Only two will be there. Rest everything will be new. So over here also you find that hybrid band is there. Rest everything which is there is the low density band, is going to be the low density band. So the question is after 60 minutes. After 60 minutes means 20 minutes more. After 20 minutes more, what do you do? If this was 2 became 4 will become what? 8. 4 will become 8, right? Yes, ma'am, it will become 8. Now out of 8, I told you 2 of them are always going to be hybrid. 2 of them are always going to be hybrid. Okay, what about the 6 of them? They are going to be the one which is going to be low density, and that is the question for you guys. The bacteria that show low density after 60 minutes are how much? 75%? How much? They are going to be 75%. Yes, Baba. Okay, yes, 6 out of 8. So basically it is going to give me 75% or 0.75, that means 75%. Got it? And 2 of them are always going to be basically of hybrid density. So if asked about 2, then you have to do like that. 2 by 8, you get your answer. You got it? And you have to be very sure about the medium that is there. Sometimes they do not change the medium. Sometimes they will ask you how many of them are going to be heavy density after 60 minutes. So heavy density is not going to be found after 60 minutes. The only time that you find heavy density is over here. Okay, if at all you're growing that over here. Got it? PDF, Baba, I have shared with you over here. PDF is already provided. Please check the pinned comment. Okay, now DNA fingerprinting. Arrange the various types of DNA fingerprinting technique in the correct order. I'm not explaining this. I'll just explain the picture. Listen to me carefully. First of all, you need to extract the DNA. You can get the DNA with the help of saliva, the hair follicle, any other skin parts and things like that. Always remember in blood also, but in blood, not in RBC. Okay, in blood you have multiple other cells. Do not take RBC, because RBC does not have a nucleus. RBC is also known as erythrocytes. Erythrocytes do not have nucleus. Okay, now you have extracted that. You have extracted that. The next thing that you do is, after isolation, is cutting with the help of restriction enzymes. You cut with the help of restriction enzymes. What do you do? What do you do after that? You do electrophoresis. After that, you have divided, you have made sure the fragments are being separated. After separating the fragments, you are going to transfer them into a nitrocellulose sheet. Okay, nitrocellulose sheet. And finally, that is known as blotting or southern blotting. And finally, you are going to put some props, complementary pairs. You want to see where are they going to attach. That is known as hybridization. So basically, what do? You are going to incubate with labeled props. VNTR props is what you call it. But anyway, you are going to hybridize with the probe, and then finally you are going to view it under autoradiography machine or X-ray machine. You will be able to see the strands which are there. That is how things are going to go. Okay, so hybridization, for hybridization you require what? VNTR probes. That is one important thing out there. Now, which of the following statement is not correct? Which of the following? There is no trick, just use your imagination. First, you take the DNA, you cut it off. DNA, you cut it off. Put it in the electrophoresis chamber, transfer it out there, and then put the VNTR props. Okay, wherever it goes and attach, I will be able to see it where? Under autogradography, that's all. Okay, next thing. Which of the following statement is not correct? A structure with many ribosomes bound to the mRNA is called as polyribosome. Suppose this is an mRNA, please always remember this is 5' end and this is 3' end. If at all you have multiple ribosomes being attached to it, do you call it polyribosome? Yes, I do call it polyribosome, correct. Now, amino acylation of tRNA requires the presence of ATP. What do you mean by amino acylation? So this is the tRNA, you have 3' end and you have the 5' end. Sorry, ulta. Okay, so you have the 5' end over here and you have 3' end over here. On the 3' end, Baba, amino acid is going to come and attach. Amino acid is going to come and attach. This is known as amino acylation, and for that you require a heavy amount of ATP. It requires heavy amount of ATP. Always remember, in the 3' end it is going to attach. 3' end, okay? So amino acylation of tRNA requires presence of ATP. Definitely it requires the presence of ATP. Okay, now next thing, exons are the DNA sequence which are not expressed and do not appear in processed RNA. Yes, it is not going to be expressed in the processed RNA, but do you think that is exons? That has to be introns, right? That has to be introns, but here it is mentioned about exons, so which is why it is wrong. Exons are going to be expressed, introns are the one that are not going to be expressed. Now, after transcription in prokaryotes, mRNA is used in the same compartment as there is no nucleus. Yes, I told you it's a coupled process. Everything is going to happen within the cell itself. Now, one very important thing, if this is the mRNA, always remember you have the start codon AUG and you have the stop codon, which is what? UAA, whatever is there. Okay, in front of that you can have UTR, untranslated region. Why do you say the 5' before the start codon you have a UTR, untranslated region? Similarly, at the end, after the 3', after the stop codon, you have the UTR. At that particular position also you have UTR. UTR stands for untranslated regions. Make sure you surely know about the 5' and 3'. 5' start codon, 3' stop codon. Do not get confused out there. UTR is there for providing stability for the initiation of the process to happen very smoothly. Okay, that's all. Okay. Yes, PDF will be there, Baba. PDF is there. This clearly mentioned. Where do you find it? You have to go to the folder, and basically in the rank booster KCET, there is biology. In biology, there is last minute revision. You can just click on that particular thing. Okay, next thing. The given figure represents the process of transcription in bacteria. Listen, in bacteria, always understand, transcription happens in what? Always. Replication, transcription, and also formation of, the protein out there. Everything happens in 5' to 3' direction. I'm talking about the new strand. Okay, the new strand formation happens in 5' to 3' direction. So your DNA should be what? In 3' to 5'. Okay, then only you will get a mRNA in 5' to 3' direction. Anyways, so this is the mRNA. Who helps in transcription? It is the RNA polymerase. Okay, it is going to be the RNA polymerase. Listen to me this question. Okay, this is the answer. So over here you have been mentioned mRNA is being formed. What is B? B is going to be RNA polymerase. What is going to be the C? C is the rho factor. Rho factor means what? Release factor. Rho means what? Release factor. So always remember, for the process of transcription to start off, RNA polymerase has to go and attach. But RNA polymerase is shy, and shy, S for sigma. So it says that sigma factor can you also come along. So initiation, you need sigma. You need sigma. Now, during termination, what is going to happen? Basically, the DNA and the RNA, they're going to move apart. They are crying because breakup is happening. Rho na is what you say in Hindi. So rho na. So rho factor is there for ending up the process of transcription. Always remember that. Similarly, over here in translation, to end up, you do not have any tRNA that will come and attach to stop codon. Rather, you will have a release factor. Release factor. So that is for translation, and over here rho factor for transcription. Always remember that. Okay. Yes, same questions are not gonna come. If at all you're looking for the same question, it is not gonna come. Concepts. That is why I'm explaining. Now in structural gene, the template DNA strand is this. Listen to me. The template, always understand. When you talk about the DNA, there is coding strand and there is template strand. The template strand is the one that undergoes transcription, not the coding strand. The coding strand will be in the direction 5' to 3'. The template will be in the direction 3' to 5'. We are going to make a new mRNA, as I said, in 5' to 3' direction on dependency of this. So in the question, if at all it's mentioned that template strand is like this, if they are mentioning coding strand, you have to make the opposite of it. You have to make the complementary of it, that is a template strand, okay, first, and then make what do you call the DNA. That is how it is going to go. Anyway, they have given you the template strand right now. You have to find what? You have to find the mRNA, the complementary, find the correct and complementary nucleotide sequence on the coding strand. So you just have to find the opposite of it. So Baba, first of all, it has to be 5' to 3'. Yeah, it has to be 5' to 3'. You mentioned that. And then wherever A is there, A base pairs with T, T base pairs with A, G base pairs with C, and that is how things are going to go. So always make sure you know the direction of it. Sometime to confuse you guys, they can mess up the question. What they can do is they will be giving you the coding strand. They will be giving you the coding strand, and they can ask you what is the mRNA that is going to be formed. What is the mRNA that is going to be formed? So how will you answer that particular question? If at all, in that particular case, just listen. If they are mentioning the coding strand is this, A, T, G, C, A, T, whatever, blah, blah, blah is there. So first thing you have to do is make a template strand. How do you make it? Make a complementary of it. So A base pairs with what? T. T base pairs with what? A. G base pairs with what? C. C base pairs with what? G. That is how you get what? The template strand. The template strand. Now you have got the template strand. Can you make a complementary of it? When you make a complementary of it, again you will get the same strand only, right? So what do you do if at all the question is like this? Always understand, they have given you coding strand and they are asking you to find the mRNA. mRNA. What you have to clearly do is use the same sequence. Wherever T is there, just replace it by U. Because if you make a complementary of it and then make a complementary of that, anyway you will get this only. But you are making an RNA, and RNA does not have thymine, it has uracil. So you have to replace T by U. You have to replace T by U. That is the logic. Okay, that is the logic that you have to use. Anyone who is going to give the examination right now, who is going to go to the examination center, first of all, wishing you the best. I want the best for you. Do not be worried. As I said, face the paper. That's all. It's just an examination. Just face it. Let's see what is going to happen. Okay, ma'am, you are my star. Thank you so much for your hard work. Thank you. And guys, don't be late to the examination center. I do not know. I am not there. That is why I am telling you. If you want to go, please go. Get inside. Yes, Jasmine. Yes, tell me. Pallavi. Okay, so anyway, wishing you the best. If you see first PUC question, get stuck there. Do not be stuck there. Skip the 10 questions out there. Move ahead with the next one. I'm telling you again. Okay, if at all you are over here listening to me, listen to me very carefully. If I see a question again stucking there, next question also I got stuck. What do? Leave those 10 questions, come with the next, start with the 11th question. You will find an easy patch out there. When you find the easy patch, it will give you confidence. Then you can come back and do these questions out there. Okay? And if at all there is a question that is taking too much time, put a star mark. If there's a question that you do not know at all, put a circle. Towards the end, you can just do random guessing. Cool? Yes. Hiyo. Next question. Find the sequence of binding of the following amino acid tRNA. You have been asked about where does the tRNA go and bind. So these are the tRNA molecules that is given to you, and they asked about where do you go and bind. tRNA goes and binds on the mRNA. Is mRNA strand given to you? If mRNA strand is given to you, let's see. So translation to mRNA, but then they have given a sequence like this. If T is there in the sequence, it is not an mRNA, it is DNA. They have given you a DNA strand. So what you have to do, you have to make the mRNA first, and then you can find the tRNA. Again, don't do the whole drama. What you have to do is just replace T by U. Just replace T by U. So UAC. UAC will, because when you make the complementary and then you have to look for the complementary, it is going to be the same only, right? In the tRNA, it is going to be UAC, that is the first one, and over here you have got the answer UAC. Cool? Yes. Now, yeah, RNA polymerase I have already explained. RNA polymerase 2 is responsible for transcription of H in RNA. I already spoke about that. Now, there's the next question that is there. A flower has 10 stamens, each having bilobed dithecous anther. Each having bilobed dithecous anther. So listen to me. Whenever you get a question like this, just make a diagram of an anther and make 4 circles over here, and then write whatever is written. A flower has 10 stamens. This is something different. Okay, keep it aside. If each microsporangium, that means this, has 5 pollen mother cells. So write 5, 5, 5, 5 over here. We know one pollen mother cell is going to give rise to what? It is going to give rise to 4 microspores, or later 4 pollen grains, right? So if 5 of them are there, so 5 into 4 it becomes 20. 5 into 4 it becomes 20. Here also 20. Here also 20. All of them will give 20, 20. 20 into 4 is what? 80. Have I got my answer? No. What is the question? They have been asking you about a flower which has 10 stamen. This is about 1 stamen. So how do you calculate for 10? You multiply it with 10, and then 800 will be the answer for this. 800 will be the answer for this. If they are not mentioning 10, it's okay. You just have to do 80 and that will be your answer. But if they are mentioning that there are these many number of stamens, you have to calculate that as well. No tension. You are going to rock it. Okay, Baba? Everyone who is seeing this particular video, you're going to rock it. Don't see my pace and get tensed. Okay? This is just for revision. I do not have an option otherwise, right? So you will rock biology examination. Go rock it. Now, there's a question like this, but before explaining that, listen to me carefully. This is an embryo sac. An embryo sac, Baba, has 3 antipodal cells, 2 synergids, 1 egg cell, and basically this is a big cell that is known as central cell. Central cell says that, central cell is a bit selfish, it says that I need 2 nucleus. So it has 2 polar nuclei. Usually, 1 nucleus will be having what content? Haploid number of DNA, haploid number of chromosome. So all of them are haploid. What about this cell? The nucleus is haploid, but it has 2 nucleus. So what happens? This cell is diploid in nature. The central cell which is there, it is the only guy which is going to be what? Diploid in nature. Now what is the question? In apple, the chromosome number of gamete is 17. The number of chromosome in the gamete, that means the egg cell, or basically all of them is going to be what? 17. If egg cell is 17, that means all of them have 17 in their nucleus. This guy has 2 nucleus, so this is 17 and 1 more 17. Okay, this guy will be having 17, here also 1 more 17, so that means it will be 24. It will be 24 numbers. It is diploid already. Anyways, what is the chromosome number in its primary endosperm nucleus? What is primary endosperm nucleus? Ma'am, you didn't talk about that at all. What is primary endosperm nucleus? So let me see. Listen to me very carefully. So over here, when pollen grain comes, okay, it carries 2 gametes. It is going to carry 2 gametes. So one gamete which is there, it is going to fuse over here with the egg cell. Another gamete which is there, it is going to fuse with what? The 2 polar nuclei. One gamete that fuses with the egg cell, okay, is haploid. Egg cell is also haploid. Haploid plus haploid, you get what? Diploid zygote. You get diploid zygote. Okay, what about this guy over here? Haploid gamete comes in. This is already diploid in nature. Haploid plus diploid, that means 2n plus n is equals to what? Triploid primary endosperm nucleus. Triploid primary endosperm nucleus. So you have been given the number of gametes. In the number of gametes, the chromosome is 17. That means haploid is 17. What is going to be there in primary endosperm nucleus? It is triploid, right? So 17, 3n is equals to what? 3 into 17, and that is going to be 51. The answer for this is going to be A. It is going to be A. Got it? Yes, fast ma'am. Okay. Now, identify the correct statement. Only one megaspore present towards the chalazal end remains functional. So, always remember megasporogenesis. Okay, megasporogenesis. If at all you talk about the ovule, you have the chalazal end and you have the micropylar end. In the chalazal end, you have the antipodal cells. In the micropylar end, you have all this thing. But before all this thing happens, always remember the formation of, the starting of megasporogenesis starts from micropylar end. Okay, now you have got 4 megaspores, and we have studied that 3 of them degenerate. Where do they degenerate? Here in the micropylar end only. 3 of them degenerate in the micropylar end. One that remains functional is going to be remaining functional in the chalazal end. Unusual thing. Usually in the chalazal end nothing useful is happening, but this time something useful is happening. The functional megaspore is there in the chalazal end, and the 3 of them that degenerates happens in what? In the micropylar end. Cool. Now, clistogamous flowers are self-pollinated because clistogamous closed flowers, they are always self-pollinated. Why? Because they are bisexual, both male and female part is there. They are closed, so they do not have any option. They do autogamy only. Okay, nothing else. So, select the plant species where emasculation is not required in artificial hybridization. Emasculation means removal of male parts. Okay, so if at all the plant is having only female flowers, do you require emasculation? No. So, that is an example. Papaya over here is unisexual plant. It will have female plant and male plant separately. So that means you do not have the male part in the female plant. It is going to be papaya. That is going to be the answer for this. Now, if we talk about the pollen grain, what is pollen grain? It is a male gametophyte. What is the embryo sac? It is a female gametophyte. Always remember, the male gametophyte is has a vegetative cell and a generative cell. The vegetative cell has irregular nucleus. The generative cell will have a elongated nucleus. The generative cell is going to revise to 2 male gametes. If at all it is dividing into 2 male gametes, how many cells are there? 3 cells. 1 this and 2 male gametes. This is found in 40% of angiosperms. They are going to release their that is pollen grain in 3 cell stage. What about the majority of them? Majority of them are going to release their pollen grains in this 2 cell stage only. What? 2 cell stage only. Everything is going on. Okay. So everyone is saying microbes in human welfare. Guys, we'll come to that. So yes, that is a scoring chapter, so I can talk about that. Just a second. Microbes in human welfare. Where is it? Okay. Yeah. Only one question is there. Yes, listen, match the following question. Microbes over here, Aspergillus niger, Clostridium botulicum, Saccharomyces cerevisiae, Trichoderma polysporum, Monascus purpureus. Just listen to me carefully. Just listen to me carefully. Hello, 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 hello. Krishna, I hope I'm audible. Listen. Three things are there, okay? Three things are there that you need to remember. Cyclosporin A, statin, and then you have streptokinase. Cyclosporin A is a immunosuppressive agent, and that is produced by a fungi. What is the name of the fungi? Trichoderma polysporum. It is an immunosuppressive agent, means if at all I am taking your kidney, my body is going to reject it. I need someone who suppresses my immune system, and that is going to be cyclosporin A. It is produced by Trichoderma polysporum, which is a fungi. You will see Trichoderma in biocontrol agents also. Please understand, it is also acting like a biocontrol agent. It kills the pest as well in the roots. It is a fungi. The next thing that we have is statin. The statin which is there, it is the one that stabilizes, ST for stabilizes, ST for statin. It stabilizes what? The blood cholesterol level, okay, by inhibiting the enzyme, by messing up with the enzyme that is responsible for production of cholesterol out there. Again, this is also a fungi that produces it. What is the name of the fungi? Monascus purpureus. Monascus purpureus. Now we have one more thing, streptokinase. What does streptokinase do? Streptokinase is a clot buster. Suppose someone is having heart attack or myocardial infarction, and basically there are clots in their blood vessels. You want to break open the clot, you have streptokinase. But this is not produced by fungi, this is produced by bacteria. The name of the bacteria is what? Streptococcus. It is what? Streptococcus. Always remember it. Suppose, Baba, okay, now the next thing that is there over here is, which among the following products of microbes is not obtained from fungi? So penicillin, you get it from fungus. Penicillin notatum. Penicillin is what? Antibiotic. It was antibiotic which works against what? Staphylococci, not streptococci. This is staphylococci, which was a bacteria. Alexander Fleming was able to get the answer for that. Discover that. Okay. Now statin is what? I told you it's from the fungus, that is Monascus purpureus. Cyclosporin A, again from the fungus, that is what? Trichoderma polysporum. What about Swiss cheese? Swiss cheese, it's produced with the help of a bacteria, Propionibacterium shermani. It produces CO2, that is why it is shermying, that is what? It is getting shy. Propionibacterium shermani for Swiss cheese, Shermani S and Swiss cheese S. Over here you have Roquefort cheese also. Roquefort cheese is not produced by bacteria, it is basically done with the help of fungi. Difference is there. Swiss cheese, bacteria. Roquefort cheese, fungi. Don't write yeast out there, it's a fungi. Okay, yeast comes under fungi, but there are multiple other type of fungi, right? So it is produced by fungi, that is according to NCERT. Now suppose you have an oily stain on the dress. How do you remove it? You remove it with the help of something that is known as lipase, because lipase is what? It's going to act on lipids, and fat is what? Lipid. Oil is what? Lipid. So you are going to use lipase. If at all you have a fruit jar over here and it's clumped up, if at all you want to clear it up, what do you use? Protease or pectinase. Protease or pectinase for fruit jars. For fruit jars. Okay. Now there is beer, whiskey, brandy and rum. Okay, beer and wine, beer and which is there, and also wine, these are produced without distillation. Without distillation means basically distillation is a process where you let go of the water content. If the water is evaporated, what is left behind? Something that is concentrated. Now if at all you have people who drink beer and stuff like that, it's okay, it does not have too much amount of alcohol. Why? It is not too much concentrated. Why? Because distillation is not happening over here. No distillation. Okay, but whereas when you talk about whiskey, brandy, rum, everything is a product of distillation. Distillation. Always remember that. I need breath. Okay, now one more very important thing, BOD. BOD is what? Biochemical oxygen demand. Baba, if at all you see, you have pollutants. If at all you see, you have pollutant. And you have what? An organism out there. That is the aerobic bacteria. Aerobic bacteria. Aerobic bacteria is going to act against the pollutants. But it says that since I'm aerobic, I need what? Oxygen. Give me oxygen. So the requirement for oxygen increases when the level of pollution is what? More. So what is this requirement known as? BOD. Biochemical oxygen demand. So demand for oxygen is more when pollutant is more. So over here in the question you have been given D1 has the highest amount of BOD. Highest amount of BOD means highest amount of pollution. If at all you see the options, which one do you think will have highest amount of pollution? Untreated sewage water. Primary effluent has undergone some treatment. Secondary effluent obviously more. River water definitely has the best one. So basically untreated sewage water has the high amount of pollutants. 5 mg per liter, that means lesser amount of BOD. BOD is less when pollutants are less. Where do you find BOD less? River water, because in river water we will only send only when the sewage is completely being treated properly. Okay, so always remember that. Who is doing all this thing? Aerobic bacteria along with what? Fungal filaments. What do you call this fungal filaments as? You will call this fungal filaments plus aerobic bacteria, you call this as what? Flux. What do you call it? Flux. Okay, now over here one more thing is there. Aspergillus niger, Clostridium butylicum, Saccharomyces cerevisiae, Trichoderma, and this is being not done, right? Aspergillus niger is a fungi. Producing what? Citric acid. Clostridium butylicum, butyric acid. Okay, you have more Acetobacter aceti, acetic acid. Saccharomyces cerevisiae, it is known as baker's yeast and also brewer's yeast. Why? Because in fermentation it produces ethanol. It is a yeast. So by Baba, this is fungus, this is fungus, this is fungus. Yeast comes under fungus, and this is what? Bacteria. Always remember that. Aspergillus niger, citric acid. Okay, got it? Hayo. Yes, one more thing. A farmer has applied chemical fertilizers in the crop field. In the next session, the crop growth was poor and soil lost the fertility. Suggest a suitable microorganism that can replenish the fertility of the soil. Baba, you can use anything, but over here, the biofertilizer which is there is only Nostoc. Nostoc is what? Nostoc is a bacteria. Yeah, Nostoc is a bacteria. It's a cyanobacteria. We have 3 cyanobacteria. One is, you say ano, you say you know, instead of you know, just put as ano. Ano is what? Anabena, Nostoc, Oscillatoria. 3 of them, they come under bacteria, but specific group of bacteria, we call it as what? Cyanobacteria. We call it as what? Cyanobacteria. They are very, very important for you. They fix nitrogen. They can prepare their own food. Okay, they can prepare their own food. They are not dependent on plant, but they fix nitrogen. Now you have normal bacteria also. What are the normal bacteria? Rhizobium is there, Azotobacter is there, Azospirillum is there. Ra. Okay, in this our rhizobium which is there, it is having symbiotic association with the plants. This is normal bacteria. It cannot prepare their own food. Basically, they have symbiotic association with the plants. They fix nitrogen for the plants. What does the plant give back? Plant gives food to it, protection to it, everything. They have a symbiotic association. What about Azospirillum and Azotobacter? They are free-living bacteria. They also fix nitrogen, but they say that I can live free. I do not have to be in association with you. That is the only thing. Cyanobacteria is different. Bacteria is different. Under bacteria comes cyanobacteria, and there's a whole different category. Apart from that, in your NCERT, there is mycorrhiza. Mycorrhiza is what? Combination of fungus and the plants. Fungus and the plants. This fungus belongs to what? This fungus, fungi and the plant root. The fungus which is there, it belongs to a genus that is known as glomus, and what does it do? It gives phosphorus. So the fungus can go inside the soil and bring out phosphorus. So it gives phosphorus, it provides resistance to diseases, it provides tolerance to different conditions, and also does one more very important thing, and that is what? Overall growth and development. Overall growth and development. What does the plant give? Plant gives food. Plant gives food. Again, this has symbiotic association out there. Always remember, in lichen it is going to be algae and the plant root. Algae and a fungi. Algae provides food to the fungi. In the case of mycorrhiza, it is going to be a plant root and a fungi. Okay, so algae and plant root, that is the thing that you have to change in lichen and this mycorrhiza. Hiyo. Yes. Guys, yeah. Now evolution chapter over here, the thing that is wrong, always remember in the experiment that he did. Okay, in the experiment that they did, first thing, it was reducing environment, no oxygen was there. Second thing, the temperature was 800 degrees Celsius. What was the temperature? 800 degrees Celsius, not 900. Okay, next thing, how many gases were there? Methane, that is CH3. Methane, that is CH4. Ammonia, that is NH3. H2O, but not N2. This is wrong. It is H2. No N2 was there, H2 was there. Okay, and no NaCl was there in the boiling water, only water was there, no NaCl. Okay, what was being formed over here in Stanley Miller's experiment? Organic compounds. What organic compound? Amino acid. Only amino acid. No RNA, no DNA, nothing. Only amino acid in Stanley Miller's experiment. Rest other people did, and they found something else also. Okay, so that is the only thing that you have to remember. Over here, the frequency of dominant allele, please listen to me. This might come, I'm not sure, but yes, so let's go. So over here, the frequency of dominant allele is 60%. Always remember Hardy-Weinberg principle, that is P plus Q is equals to 1. Over here, you need to know dominant allele is represented by P. And what are you saying? The dominant allele which is there, it is 60%. I can say 60%, I can put into 0.6. I can convert into 0.6. Now, what I have to find? I have to find the percentage of heterozygous individuals. That means organism that looks like this. I'm not talking about about homozygous dominant, I'm not talking about homozygous recessive, I'm talking about the percentage of them, okay, like capital A and small a. How do I find that? I need to find the dominant allele frequency that is already there. I have to find what? The recessive allele frequency. Then only I can do something. So I have got the dominant allele frequency. Can I find the recessive allele? Yes. How? By replacing it. So listen, P is 0.6 plus Q is equals to 1, right? Now Q is equals to what? 1 minus 0.6. Q is equals to what? 0.4. I have got 0.4. So ma'am, I have got A's value and I have got small a's value. I'll just do multiplication. I'll get 0.24 and that will be the answer, right? 24% wrong. Always remember, when you're talking about heterozygous individual, okay, when you're talking about heterozygous individual, things are going to be different. It is going to look like this. There is a formula, P square plus 2PQ plus Q square is equals to 1. P square is what? Capital A square, that means AA. Whenever you want to find homozygous individuals, you just multiply this, okay, 0.6 into 0.6. But when talking about heterozygous, 2PQ, 2 into P is what? 0.6 into 4, PQ is what? 0.4. Your answer will be 0.24 into 2, that is 0.48, that is 48%. Do not forget to multiply with this 2 out there. Very, very important. Okay, so the answer would be this. Whenever you get these type of questions, you have to go a bit slow. You have to go a bit slow. I see Malayalis out there. Hello. Okay. Okay, cool. Now the next thing that is there is match column 1 with column 2 and select the correct option from the given quotes. Genetic equilibrium, gene flow, saltation, genetic drift. First of all, Hardy-Weinberg said that the equilibrium is going to stay remaining same throughout the generation, but there are some factors which denies it. For example, gene flow. I cannot say it is going to be remain the same. For example, you are there, now you come over here. The allelic frequency over here is going to change, right? Emigration is going to change up the allelic frequency. So gene flow is going to be changed by emigration, change of allele. That is gene flow. Okay. Genetic equilibrium means it is going to remain same throughout the generation of throughout generation to generation. Genetic drift means what? Genetic drift means, for example, Baba, over here you have a population, and I am saying, by the way, everyone who is watching this video, okay, you have to come back and tell me how was the examination later on. And do tell me if at all this particular video helped you in some ways. Obviously, I have not completed everything. I cannot do that, but yes. Anyways, so suppose here you had yellow color beetles, okay, and you had green ones later on. The yellow one said that green ones, you get out, you cannot be here, okay, they were dominating over here. Now what happened? One day an elephant came and stamped over it and killed all the yellow ones. Did the allelic frequency change? Yes. Did it occur by chance? Yes. So this is known as genetic drift, when the allelic frequency is going to change by chance alone. Very, very random process that is going to happen. Saltation is what? Saltation means single step large mutation. One single step can lead to large mutation. One single large mutation can lead to what? Evolution. That is what you mean by saltation. Hugo de Vries spoke about that. Always remember, Charles Darwin was there and Hugo de Vries was there. Charles Darwin said that evolution which is going to happen is going to be a slow process. It happens slowly and things like that. Whereas on the other hand, Hugo de Vries said that it is going to be really, really fast and it is going to be random process. Both of them are equally correct in their own ways. Okay, there's a guy whose name is Thomas Malthus. Thomas Malthus was the one who influenced Charles Darwin, not Lamarck, not anyone. Thomas Malthus and Charles Darwin, friends. Always remember that. Okay. Yes. Now there is a question like this. The image is given over here. You have to find what is happening in either of these cases. Always remember, Baba, always remember one very important thing. If at all you have capital T and small t, okay, if at all you have capital T and small t in a generation, frequency of allele is there. You are saying extremely tall people are there, extremely dwarf people are there. Then you are saying that no, in this time you have got someone with intermediate height. That means the middle character is being selected. This type of selection where the intermediate character, middle character is being selected, you call this as what? Stabilizing selection. What do you call it? Stabilizing selection. Okay, the next one is where, suppose again tall and dwarf is there. I am saying only dwarf ones are selected. There is a written peak shifts towards one direction. Either of them, one of them is going to be selected. Okay, just read. If at all you are not able to understand the difference between this, read in one direction. That means only one guy is going to be selected. That is known as directional selection. Okay, now disruptive is there, where you are saying that extremely tall people are there, extremely dwarf people are also there. Both of them are there. That means it is going to be disruptive selection. It is going to be what? Disruptive selection. Always remember that as well. Cool? Yes. Hiyo. The brain capacity of just a second. Okay, the brain capacity over here, didn't I bring it? Oh my god. Okay, so brain capacity, Baba, listen to me very carefully, everyone. If at all it could be helpful for you, you can just listen to me. There are two man, okay, man. One is Dryopithecus and one is Ramapithecus. Dryopithecus more ape-like, Ramapithecus, Rama, more man-like. Rama so sounds like a man, right? Okay, both of them 15 million years ago, both of them are going to be, so someone said draw hence is a way to remember. Draw hence. Okay, draw hence is what? Dryopithecus, Ramapithecus, Australopithecus, Homo habilis, Homo erectus, Neanderthal man, and Homo sapiens. Okay, so over here, just give me a second. Just give me a second, okay? Yes, by the way, you guys don't have to go. It's 1:33 already. Okay, anyways, so over here, both of them 15 million years ago, this was more man-like. This was more man-like. Australopithecus, the unique feature about them is that they were found in East African grasslands. East African grasslands. Okay, please remember that. Okay, East African grasslands, and also they ate only fruits. They found weapons and all, but only for eating fruits. What about H? H stands for Homo habilis. The first Habibi, first human-like features were found here. They are what? Homo habilis. Homo habilis, very important. First hominid features were found over here, and they also ate only fruit. Another unique feature about them is from here you have got the cranial capacity. What is the cranial capacity? It is 650 cc to 800 cc. That is the first cranial capacity ever mentioned. Then you got Homo erectus, the one who could stand erect. Homo erectus. And from this point onwards, things are going to change. They are going to start what? Meat. They started eating meat, and their cranial capacity also increased. What is it now? It was 650 to 800 cc, but now it is 900 cc. Always remember, they are also known as Java men. Homo erectus is also known as Java men because their fossils were found in Java, Indonesia. Their fossils were found in Java, Indonesia. Now we have Neanderthal man. Neanderthal man is basically the most important thing. Neanderthal man were having 1400 cc. 1400 cc cranial capacity, the highest of all of them. They buried their dead and also covered their body. They buried their dead and they covered their body. And then you have got Homo sapiens, that is us. Okay, now talking about the years, always remember this was 15 million years ago. Over here, when you talk about Homo erectus 1.5 million years ago. And Neanderthal man, almost 1 lakh. Okay, 1 lakh to 40,000 years ago. And in between that, you have got sapiens also, Homo sapiens, that is 75,000 to 10,000 years ago. So basically Homo erectus was there, it formed Neanderthal man, but parallelly Homo sapiens, that is us, were also forming. Nothing about us is being given as in what is the cranial capacity and things like that. Okay, this is very important. I hope you have got it. Yes, guys, everyone who is there, please be on time. You can just... Yes, please, 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 please go. Those who has to go, please go. Everyone who has to go, please go. And everyone who have not hit the like button, click on the like button right now. Okay, before going, only if you wish to. Only if you wish to. Let's make it 500. Anyways, that's not a concern right now. Anyways, follow. Please go, give your best. That's all. And those who are staying, those who are somewhere near, you can stay back. Ma'am, do tough question. What do you mean by tough questions? This is a question that came, right? So we have to do that. Again, some scoring chapter I'll try to do so that it becomes easy for you to get marks at least. Where is reproductive health? Reproductive health. Yes, this is reproductive health. So listen, in reproductive health, there is natural method which can... Thank you. Okay. Okay guys, how many of them are staying back? You need to go to examination hall, right? 2:30 is the paper. Thank you so much, ma'am. Please take care of us. Have enough water. Take care of your hats off you. I love you there. Thank you so much, Harley. Means a lot. Thank you, Jasmine. Thank you so much. Can still till 2. Okay, then I'll continue for those who are there. Yeah, and when the number goes down, I'll stop it. But anyone who is there and who have to reach the center, make sure you go fast. You do not stay over here, please, 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 please. Okay, Okay, fine, cool. So dash method works in the principle of avoiding chances of ovum and the sperm meeting. What is that? Basically, it can be, okay, the one that avoids chances of ovum and the sperm meeting, it can be barrier method, it can be natural method. Anyways, A is going to be natural, because the other options, if at all you see, you'll get to understand. But anyway, one such method in which couples avoid or abstain from coitus from day 10 to day 17. Always remember, day 10 to day 17 is what? The fertile period. It is what? The fertile period. It is the fertile period out there. So even if at this particular point of time, if someone has coitus, there are high chances that baby can be formed. Okay, baby can be formed. So basically, people abstain from it. People they are absent during this particular period. That is known as periodic abstinence. Okay. Next thing, that is another method in which male partner withdraws his penis before ejaculation to avoid insemination. That is known as withdrawal method or coitus interruptus. You are interrupting between the coitus. That is why coitus interruptus. Finally, there is a method which is based on the fact that ovulation and therefore cycle do not occur during the period of intense lactation. What do you mean by that? What do you mean by that? Intense lactation is a period where the mother is breastfeeding the child, right? Okay, so for that you need a hormone prolactin. When prolactin hormone is high, ovulatory hormone, that is FSH and LH, they cannot be high. So it stops it, which is why ovulation is not going to happen, which is why menstruation also not happens. But this is highly risky. Anyways, this process is known as lactational amenorrhea. Amenorrhea means no menstruation. No ovulation means no baby also, but this is highly risky. All the natural methods are risky. Please remember that. The answer for this question is going to be this. The next question. A childless couple visited Assisted Reproductive Technology Center to get assistance to have a child. On diagnosis, it was noticed that there was low sperm count. Just directly think your brain into direction of for what? The male methods, that is artificial insemination or IUI, things like that. Now, which of the following strategy of ART is most suitable in this case? Gamete intrafallopian, no, this is for females usually. Zygote, no. In vitro fertilization does not count over here. Artificial insemination is given here, right? Artificial insemination means artificially you are going to inseminate. Artificially you are going to put the sperm inside the female reproductive tract, that is known as artificial insemination. Okay. Now, one very important thing, just a second guys, for those of you who are going, because I see lots of thank you. God bless you all. Okay, do good. You will do good. Do not be worried. You are going to rock biology examination. However shitty the paper is, you will rock it with the amount of knowledge that you have. You can only do what you can and you will do good. Okay, bye. Lots of love from Jashna. Okay, for those who are gone. For those who are going to go, this is for you as well. Anyways, listen to me. If at all you are putting semen inside the uterus, you have another term for it, and that is what? IUI, intrauterine insemination. It is what? IUI, if you are putting inside the uterus. If not, if you're just mentioning that you are putting anywhere inside the reproductive tract, it is known as artificial insemination. Okay, the next question. The government of India legalized MTP with some strict regulation in the year. MTP was legalized in the year 1971. In the year 1971. Okay. Now listen to me very carefully. When it was legalized, people started doing it. Some unqualified people also started doing it. For example, if someone of your age is pregnant, they go to the doctor. Some unqualified person, they will do it. There are chances that it can harm the girl who is there and also the baby. They can be killed as well due to what? Unethical methods that are being applied over there. Which is why there were a lot of deaths being reported, and that is exactly why there was an amendment act being put. Even though it was legalized in 1971 plus 1 you do, it becomes 20, and here you do ulta. 2017 you got MTP, Amendment Act. Amendment Act means more strict regulations were being put down, according to which a person can undergo MTP, medical termination of pregnancy, in 3 months and also in 6 months. But if they are doing in 3 months, they need to take permission from one qualified registered medical practitioner. If at all they are doing in 6 months, they need permission from 2 medical practitioners. Always remember, 3 months means how many weeks? 1 month has 4 weeks. 3 months means 3 into 4 is equals to what? 12 weeks. 6 months means what? How many weeks? 6 into 4, 24 weeks. Okay, 24 weeks. These things are very, very important. Ma'am, up to what time is it safe? If that question is coming, up to what time MTP is safe? Only up to 3 months. Until what time it can be done? Up to 6 months. So it is safe only up to 3 months, but yes, you can do it until 6 months. Until 6 months. That is the logic behind it. Okay. Since we are talking about years, I'll talk about one more year, 1951. Family planning was introduced in 1951, and basically they came up also with one thing, that is RCH, Reproductive and Child Healthcare Program. Reproductive and Child Healthcare Program. That is also something that you have to remember. Now, in IVF treatment, in vitro fertilization, if at all you are doing in vitro, that means you are doing in the laboratory, you're fertilizing, you've got the sperm, you've got the egg, and you're fertilizing it. That is known as in vitro. In vitro means in the lab. My stomach is paining. Okay, anyways, so after you do that, what is going to happen? You are going to get the zygote. Yeah, it can be zygote if it is single celled. It can be an embryo if it is more than one cell, right? So if at all, that is a question, that after IVF, if embryo is having 8 or less number of blastomeres, then it is transferred to A. While if the number of blastomeres is more than 8, it is transferred to B. What is that? So if it is less than 8, Baba, you are going to transfer it in the fallopian tube. If it is more than 8 blastomeres, you are going to transfer it where? You are going to transfer it inside the uterus. Always remember. Otherwise it can lead to ectopic pregnancy in different area. Okay, so always remember that this is known as ZIFT, zygote intrafallopian transfer, where it's not just zygote but also the embryo up to 8 blastomeres. And this is known as IUT, not IUI. This is IUT, intrauterine transfer, where what are you doing? You are putting the embryo more than 8 blastomeres inside the uterus. Okay, and then there is gamete intrafallopian transfer, GIFT, where what are you doing? If suppose you are not able to produce enough good quality of ovum out there, you take it from the donor, but you are able to provide facility for fertilization, for normal fertilization to occur. So what do? Basically, you take the gamete and you put it inside the fallopian tube. And here in this particular case, always remember, it's not in vitro, it's going to be in vivo. That means inside the body, fertilization is going to happen. Similarly, in ART also, artificial insemination also, you are putting the semen inside. No, fertilization is going to happen inside only. Only it happens outside in ZIFT as well as IUT. ZIFT as well as IUT. Okay. Yes. Okay. Now restriction enzyme. This question anyway we'll do. Guys, there is IUD. I hope you know IUD, intrauterine devices and stuff like that. There are 3 types of IUD. If I go talk about that, it won't be finished, so I have to move ahead. Any important stuff from biomolecules? That GLUT4 thing and insulin thing is very important. Apart from that, there was one more thing I forgot. What was that? The enzyme activation energy is less if at all you have enzymes been involved. If enzyme is not been involved, you need more activation energy for the process to start up, right? Okay, guys, then I'll stop it over here. Last 3 chapters. Oh my god, still there. Okay, by the way, I'll just talk about this also once. Eco R1 has a restriction site 5-GA-TC, GAA-TTC, but it makes a cut between G and A. So please know about the direction of it. It is 5-3-, and also the cut is between G and A. Cool. Now the last 3 chapters, I do not know, guys. It won't be finished. Go. It will take a lot of time. Okay. One more thing. Over here, alpha-1 antitrypsin is for treating emphysema respiratory disorder. Rosie the cow has which milk? Alpha-lactalbumin, the human milk. Okay, the human protein which is there, alpha-lactalbumin in Rosie the cow. Okay, now you have been asking about the final chapter out there, so I think I can talk about this first. Okay, this is a pyramid, age pyramid. Okay, if at all you see the lower portion is less, that means pre-reproductive age group organisms are less. If they are less, that means in future there are no people to reproduce. That means the population is going to decline. If at all this is large and everything else is small, like a upright pyramid, in that particular case we say it is expanding. Why? Because they will be producing children in future, and that's how it is going to expand. Now you have one more thing. Stable is where pre-reproductive and reproductive are going to be in the same level itself. Now over here, monarch butterfly feeds on the poisonous meats during its caterpillar stage. Do you think that is correct? Definitely correct. Why? Because it wants to be distasteful so that no predators can eat it. That is the only only reason why it does that. Okay, that is a predatory mechanism. Now, which other pollen does not pair show commensalism? Okay, orchid and the mango. Orchid and what is commensalism? One guy is being benefited, other guy is neither getting harmed nor benefited. So over here, mango is the one that is neither getting benefited or harmed. Orchid is the one that gets benefited. Why? The orchid grows on the mango tree. It does not harm the mango tree. It just says that I need sunlight. You just allow me to be over here. So it is getting benefited. Cattle egret and grazing cattle. Again, this is neither benefited nor harmed. What about cattle egret? Cattle egret, basically when the cow moves around, it makes sure that the insect goes up, and the cattle egret basically eats it. So this guy is getting benefit. Now sea anemone and the clownfish. Clownfish is there, sea anemone is there. Sea anemone have tentacles, and in between that the clownfish remains. So clownfish is going to get benefit. What about the sea anemone? It's neither getting harmed nor benefited. So this is also an example. Now glomus fungi and the plant root, this is an example of mutualism. Both of them are going to help each other. One more example of commensalism is there, and that is whale and the barnacle. Always remember, the whale is very big, right? So and the barnacles were very, very small. On the surface of the whale, barnacles go and sit. Why? For transportation. Is the whale getting harmed? Whale is not getting harmed. Barnacle is getting benefit of it? Yes, definitely. Similarly, you hear about barnacle over here in competition, and that barnacle is balanus and thalamus. Balanus and thalamus. Always remember, balanus, B comes before C, so B is the one that is superior. If at all balanus is there, it asks the thalamus guys to move away from here and says that I am going to occupy the whole region. This is known as competitive release. When some other guy is there, the inferior guy moves away. Now the question is, if at all I am removing balanus, I am removing the balanus over here, what is going to happen? Basically the thalamus will be very happy. It is going to flourish, right? It is going to increase the number. So basically if balanus is removed, thalamus population will be flourishing. Why? Because you have removing the superior variety. Yes. And over here, the last one, parasitism, amensalism, competition and mutualism is given. Mutualism is where both of them are going to benefited. So I'll write both get benefited. Competition is where both of them are going to be harmed. Yes, minus relation is competition. Both of them are going to get harmed in some or the other way. Parasitism is where one gets harmed, another gets benefited. Okay. And amensalism is where one gets harmed and the other one is neither benefited nor harmed. So it is -0, just opposite of commensalism out there. Okay. Ma'am, which month hair on the body and the head? Which month hair on the body and the head? So listen to me. If at all you want to know about that, I can talk about it. So anyways, whatever you ask me, I can do that as of now because it's you. So anyways, this is how it is going to go. First month, which is their heart, is going to be formed. Whenever someone dies, the first thing that you check is heartbeat. Similarly, first month, heart. Second month, limbs and digits. Limbs and digits. Third month, first trimester, what is going to happen? Most of your organ system, everything is going to be formed. External genital organ is also going to be formed. Third month is known as first trimester as well. Fourth month, skimp mardi. Fifth month, baby says that God give me hair and give me movement. So first movement and first hair, FF. Okay, and the fifth month. Okay, the fifth month is where first movement and the hair is found on the head, not on the body. Now God said in the 6th month that you wanted hair. No, have hair on the body also. So in the 6th month they have hair on the body. Eyelids are going to be separated. Eyelashes are also going to be formed. Okay. Now guys, I think we should stop. I think we should be stopping, isn't it? Yeah. Have some break because we're going to face the examination. Take a little amount of break. And yes, ma'am, Pakka strategy, I made a separate video on that. Go watch it if at all you want. There's a separate video on the channel for Pakka strategy. Oh my god. No, what no? Okay, it's okay, continue. Okay, fine. So when it becomes 0, that's when I have to stop. Okay, fine. So guys, what is the next thing? Secretory phase, I cannot explain now. Okay, this I can try to explain. The correct sequence of sperm transport is what? Seminiferous tubules, then radiotestis, vasa efferentia, epididymis, vas deferens. So Ravi visits every village. Okay, Ravi visits every village and that opens into ejaculatory duct. That is how it is going to go. That is the only way I can tell. I can come to something easy that can be giving you marks. Yes, hormones, HCG, HPL, okay, HCG and HPL, human placental lactogen and human, chorionic gonadotropin, both of them are released by placenta. What about relaxin? Relaxin is released by ovary. Relaxin is released by ovary, always remember. Okay, what about this? Progesterone and androgens. Progesterone is released by corpus luteum, and Baba over here, androgens is released by Leydig cell. Always remember, placental hormones are 4 in number: HCG, HPL, progesterone, estrogen. Ma'am, progesterone, estrogen are released by ovary also, but during pregnancy it is by placenta. Okay, what does the ovary do? Relaxin. Relaxin makes the ligaments relax. Okay, ma'am, there is one more hormone, oxytocin. Yes, oxytocin is there for parturition, for contraction. Okay, and oxytocin basically is released by posterior pituitary. Posterior pituitary, not anterior. Anterior one is the one releases FSH and LH. Posterior pituitary, P for posterior and P for parturition, that produces oxytocin. Always remember. Okay, so that is the logic behind it. Now always remember, when you get a primary spermatocyte, from the primary spermatocyte you get 4 sperms, but from the primary oocyte you only get 1 egg. So 4:1 ratio is always there, and you have to make sure about the ratio. If spermatozoa and ova is given like this, so spermatozoa 4, ova 1. If ulta is given, then 1:4. So you have to be know about this particular thing also. Shobha, I already spoke about the first PUC data. Okay, I've already completed. The first thing that I did was that. Now over here, mammary gland, if at all those type of question comes, just understand the logic. Mat and dal. Mat and dal. What is mat? Mammary lobes. Mammary lobes are there. They have alveoli. Alveoli opens into what? Tubules. Tubules forms what? Duct. Ducts opens into what? Ampulla. Ampulla opens into what? Lactiferous duct. So A is twice. So please understand, here after A it is T. Okay, in ampulla. Okay, after A it is L. So lactiferous duct. Dal fell on mat. Put mat first and then dal. You will get your answer. M stands for mammary lobes over here. Do not get confused. Okay, do not get confused. Okay, mammary lobes. And yes, that is the logic that you have to apply over here. Rest everything, Baba. The things that I can try to explain, I'll explain over here. The most infectious fatal type of malaria is what? Plasmodium falciparum. It is what? Plasmodium falciparum. Cool. Yes, and injection of, if at all you get snake bite, okay, and you are getting ready-made antibodies, what do you call this? Is passive immunity, artificial passive immunity, because you're getting from outside. Antibodies are coming from outside. Got it? Allergy, during allergy, which is the antibody released? IgE, allergy, IgE. Who is, what does it do? IgE is there on the surface of mast cells, and mast cells, Baba, releases what? Mast cells are going to release histamine and serotonin. Histamine and serotonin. Both of them are going to lead to vasodilation, blood vessel dilation. How do you treat it? You are going to treat it with the help of what? With the help of steroids, adrenaline and antihistamine. Please remember, steroids, adrenaline and antihistamine. Okay, apart from that, morphine is what? An opioid. Okay, from morphine you get heroin. Morphine is a sedative and a painkiller. Sedative and a painkiller. It makes you sleep. Okay, and yes, what else is there? You get it from opium poppy or Papaver somniferum, and it is given to people who have underwent surgery. This is how you can remember. There is nothing else that I can talk about right now. Barbiturates, BAB. Always remember, BAB. Barbiturates, amphetamines, benzodiazepines. They are there for treatment of depression. Okay, they are for treatment of depression as well as insomnia. If you're not able to sleep or things like that, BAB comes to support you. BAB. Always remember. Cannabinoids such as things that you get from cannabis sativa, they are going to affect your central nervous system. And always not central nervous system, not remember it. This is cardiovascular system. Cardiovascular system. Okay, cannabinoids, the only one that affects your cardiovascular system. Example is marijuana, ganja and all those things. And one more very important thing that you need to remember is that it is the one that is abused by sports people. Sports people, they use it more. That is why abused. Okay, now cocaine, which is there, cocaine is not given to the patients. Cocaine is something that you get from Erythrosylum coca. They come under coca alkaloids, and Baba, they are they basically mess up with the CNS, central nervous system, and especially the dopamine transport. So dopamine is very important for you to be happy. They mess up with the transport. It makes you even more happier. Okay, you get sense of euphoria and also hallucination. There are three things that causes hallucination. One is etrazolam coca, that gives cocaine. One is datura, and one is atropa belladonna. Three things. Okay, what is it? Etrazolam coca, datura, which is there, and also atropa belladonna. EDA. Remember whatever you want to remember. EDA in Malayalam, EDA. Anyway. Okay, so this is that. Please make sure you know about that as well. Rest everything. One more thing which increases the blood pressure, it is going to be nicotine. Okay, and yeah, guys, I do not know how will be the paper. I just wish you the best. So that's all. Signing off for now, because I think everyone has gone. Wishing you the best. Please take care of yourself, and lots of love to all of you. You will do good. Just pray to your God and give your best, and relax. Cool? Okay? You hit such beautiful soul. Thank you, Jai Prakash. Okay. Hi, diseases. But one more thing, cancer can be found out with the help of, if you want to find the cancer of internal organs, what do you have? You have MRI, computer tomography, and radiography. Okay, MRI, computer tomography, that is CT scan, and also radiography. Apart from that, you cannot find it. Cool. And yes, Dhruv, thanks for your support from Harvest. Oh, that's so sweet. Okay, bye-bye. Wishing you the best. Thank you so much, Akshaya. Everyone, go give the examination now. No study. Hello, Alfina and Trichulika. Okay, Trichulika. Malayvilla. Parturition, dear. Parturition is induced by the fetal ejection mechanism, okay, where both the fetus and the placenta gives the first signal, okay, fetal ejection reflex, and then further oxytocin takes over the role. Okay, so guys, bye-bye. I'm going to sign off. Wishing you the best. Lots of love. Bye, Jivita. Bye, everyone. Best wishes. Noble stone, real ekdam.